information.
बोलिए सर, बोलिए.
Yes. Please, once again, I am requested all participants, please join. Our speaker is also online. Okay, sir, I will start. Okay. Now, may I introduce our eminent one speaker, that is Dr. S.P. Ahlawat. He is a principal scientist working in a division of plant exploration and germplasm collection. And he is a team leader in UNEP GEF project. It is a mainstream of management agrobiodiversity, conservation and utilization in India and other geographical regions. And he is a team leader in augmenting PGR in between Uzbekistan and India. And he conducted more than 30 explorations as a team leader and nearly 1,000 accessions he collected from different agrobiodiversity hotspots. And as a team leader or nodal officer, he is a TSP and NEH, SCSP and other mega projects. And very good expertise in collection and conservation of germplasm. And he reported new species, 4 new species, and new records or uses or any ITK, more than 50 he is registered, and 2 genetic stocks are registered. And his vast experience as a review in different national and international journals. And he published more than 112 research papers and the prestigious Brandis Award, Brandis Prize by FRI Dehradun. And he received that award. And he is a main key person in the Borjali conservation of wild germplasm of rice. And government also notified, the Assam Biodiversity State Department also recommended, and government of Assam, they recommended a biodiversity heritage spot. Actually, it is a state biodiversity. And it is a really, it is a very achievable and record approach by the speaker. And sir, now I am inviting you, and please kindly share your vast expertise, knowledge on the collection principles and procedures. And it is a very important step in the germplasm procurement, and it is a basic and fundamental step. And sir, please kindly proceed and kindly give your start your presentation, sir.
Okay, thank you, Ravi. Please share your.
Yes, sir.
May I share my screen? Is it visible?
Yes, sir. It is visible, sir.
Okay, it has come. So may I move to the next slide?
Yes, sir. It is moving, sir. Yes.
So, good morning to all. The lecture is on principles and methods of the plant genetic resources conservation. So this is a basic activity, first activity. Starts with exploration.
सर आपकी आवाज एक मिनट.
Okay. Is it audible now? So this is the first activity.
Yes, sir. Sir, continue, sir. Yes.
So is it audible now?
Yes, sir. Audible, sir.
So this first activity, everything starts from the exploration and collection. And when we talk of the plant genetic resources, consist biodiversity, that is a totality of genes and ecosystem level, that we deal with the genetic diversity at the genetic variation level and ecosystem levels. There are several around 60 slides, so I will go very fast. It will not take much time. Because it's a slide, will take some time. So coming to the why we need to collect the germ plant. So you know very well that there are for 250,000 plant species on Earth. Plant is unpredictable and dynamic because it varies with time. Here it was yield, quality, then abiotic stress, so it's dynamic. And germplasm, we have to collect the habitat loss due to anthropogenic activities, development, agricultural area lands are decreasing, then forests are also decreasing. There are danger of loss of genetic resources, and we need to conserve that, explore, and diversity missing or if insufficiently represented or gene banks, then users' needs expressed at national international levels, the users' need. Then we have to conduct the rescue of collection also, where there is an imminent threat, natural calamity, or immediate use by the local community. So, with respect to the rescue affected areas, Andaman Nicobar Island, Sardar Sarovar, these are the things which we need to correct. So these are the again why there is unpredictable dynamic demand, and for which we need to prioritization at species level and geographic regions. And clear need exists by expressed by users. So they are center of origin. It's Law 1935 developed the theory of center-based center of origin parts, and they are domesticated for having the maximum center, maximum diversity. So there are 8 centers of the diversity. Among them, Hindustan is center of that. Burma. That's also one of the diversity. We are dealing with that. So this is about the Indian diversity, genetic resources. There are 18,500 species of plants, 816 wild species, varieties of the crop plants, and 1,500 edible plants, and 583 crop plant species which are cultivated. The important one, 168 crop plants. Business to this is a large number of species, crops which we have to deal. So this Indian subcontinent has been divided into 10 biogeographic zones based on distribution of flora. So it's not possible to describe here each and everything. This is the Himalaya, Gangetic Plains. These are the different regions. This and we have to target these areas, these particular crops. Then what is the importance of the germplasm? No, there are several varieties have been directly released for the selection, particularly in perennial tree species, particularly fruit tree species, perennial. So in mango, dasari, langda, chosa, malda, etc. Anamati. These are the things, several varieties. More than 100 genetic stock species have been identified. The plant itself are directly used. So coming to the some points of wild relatives, crop wild relatives. You know, these agriculture started 10,000 years ago, and the first purpose gathered the seeds started showing them domesticated it. So the crops what we are growing now, cultivating now, they have been domesticated and brought into this condition from the wild conditions. So there are certain other species which are related. Relation means close relation, like brother, sister, like that, parents, such like that. So they also possess genes for adaptability and for disease pest resistance. They are very important. They are needed, much more needed than ever before. They challenge climate, etc. But they are under threat from massive unprecedented around the world. So this is the definition. So that also for 861 slides. That also we need a voice. So coming to the wild relatives and status, what are their status? What we are prioritized. So there are 741 genera, 833 species, and we have grouped in into 13, 12 crop groups like cereals, millets, pseudo cereals, grain, legumes, etc. And 168 crops. These are the relatives of 168 crops. So number of species are given here. So coming to the conservation part, that 13,400 accessions, 1106 wild relatives, wild species of plant genetics in the gene. This is quite low in number. So with respect to prioritization, for the 30 species of 274 have been prioritized. Wild relatives, 50 important crops from this degree. And again, we have further narrowed down to high priorities. Very close taxa having traits of utmost need, which is our prime target. There is another book also we have published, on which we have given the detailed account of the services. So coming to the fruit tree species, so I have given a, for example, important wild species. These are the indigenous, mostly indigenous species, and what are the cultivated species and their wild species. A list of them which we have to target are important for exploration, collection. Those fruit trees and they are all more than that. So these are the, we have identified the target area also. 7 areas which are the high priority area. There are chances of getting good diversity number of that species across the country. So starting from the Himalayan region, West Himalayan Gangetic plains, like different type of species. These are the target Andaman Nicobar. These are the areas. There is a problem. How to remove that? I don't know. The problem. So coming to the partners from the institutes which are taking part in this training program, so they also need germplasm. So many times we receive that request for exploration and collection, but for that, who want to collect, but first they should identify their related needs, check for availability in National Green Bank, and ask correspondence with germplasm. If it is available, then indent for germplasm. No, then send the proposal for exploration. So we prepare 5-year plan in advance. So in urgent cases, the immediate need can be urgent demand can be set. NBPGR for exploration collection. So NBPGR is a nodal institute for the plant database, and the purpose is to avoid duplicate collection. Many scientists here and others, they independently go on explorations without assessing the whether it has been already collected or not. They send the germplasm to us again for IC number. This NBPGR is kept in dark. When we receive the germplasm, then we say no, it's duplicate material. We have already collected, so we don't assign the IC number to that material. Please don't do such activity. Please check the database, which I will given. अभी ये मेरे इसमें थोड़ा ये हो गया प्रेजेंटेशन पढ़ना लाइन आ रही है so I don't know how it happened.
Sir,
So,
look at,
No
Add
need
lines.
for, no,
I
no
did
need
it,
for
huh?
remove, sir. It is okay, sir. There is no problem. There is no problem, sir.
no problem, sir. Sir, you carry
Okay,
on.
okay. I continue.
Yes, you continue,
Okay.
sir. Yes.
Okay, okay. I don't know how it came up.
Yes.
So this is a systematic plant exploration collection which started in India in 1946. In the Imperial Agriculture Research Australia. Dr. Harbhajan Singh, the eminent pioneer at plant exploration, which was initiated this activity, and he is considered the Babylon of India for this plant activity. It was important to tell you about how they are conducted. So coming to the explorations, that's to look something with intention and to utilize it. So it involves exploration has three main phases divided into three active parts. First is the planning, exploration mission, prioritizing the species, area, period, etc. That will be slides, and collecting procedures, recording information, etc. This presentation deals in three parts. So first planning, then collecting procedure, recording, and post collection handling. So coming to the planning part, and mainly we conduct for the three purposes: immediate need for use in breeding program and for evaluation, conservation, genetic diversity study, taxonomy, phylogenetic, and biosystem. General purpose is to have maximum genetic representation of population without damage to the population. That we come into the sampling part. But important is that we have to take care that without damage the two populations. Just because RET, rare endangered threatened species, accepting this question. These are traits are highly heritable, then phenotypic selection is effective, like fruit, bird, safety. Breeders know very well. That these days, our purpose to identify close relatives of crops, protecting species. So there are three types of collection missions. First is multi-crop exploration mission, that's very specific exploration. Several crops or almost all, and we go for crop specific missions also. That covers larger area. And rescue missions have been conducted where these are the rescue missions area affected and that's covered. So Sardar Sarovar Dam, catchment area, etc. These are the rescue missions have been given over here. Okay. So, but there are some diversity rich areas also have been covered. In Assam, Rohit, flowers, here, National Tiger Reserve, like that. Then earlier, prior to the CPD, international missions were also conducted involving other countries. As with Indonesia, with USSR, with Mali and Nigeria, with FAO, IPGR, like that. But now it is not, these are not being conducted. I have a project directly with Uzbekistan, which we are exchanging the journal. Bilateral vision. So this is about the status. So far, more than 2,800 explorations we have conducted till 2021. And more than 250,000 accessions have been collected in these explorations. That includes more than 211,000 cultivated and 38,000, 39,000 wild relatives also. This is the share of the wild relatives now. Earlier it was around 14-15%. 7%. Our focus is now on the number of wild. So among the collected accessions also, I had shown here the maximum ratio is the cereals followed by pulses, then vegetables, then spices, condiments like that. And this is a activity when in 1976 NPPGR was established. So every year, how much explorations have been conducted is there given over here, and total collections are given here. And the crop wild natives cultivated up to 2021. This is a period from 99 to 2005 that NAPP project was going on, where more than 1 lakh, nearly 1 lakh accessions have been involving all the crop-based institute, SAUs, and several NGOs. The phase period where the maximum was exploration. So coming to the exploration activity, exploration plan, what we have to do, we prepare a plan well in advance, which is released by the end of the February, so that 1 month before we can submit the to the activity in the first week of the April. So what we prepare the plan we give the species, etc., like wild species, big umbrella, palensis. We give the we identify the states, like, and period, then who will be the king leader, who will be the. So when you are sending us the proposal, it should be in this format. You many times the people, our colleagues from the institutes, they send the proposal several states without identifying the district. So it's not possible to cover too much of the area in one plan. This is very important. So coming while I was saying that there are so many 168 the where indigenous crops, 67 species, important species. So more than 1,500 species are there. So we have to prioritize them. Like we had done the profile relatives. We came down, narrowed down to the 100 from 860 species. Like this, we have to prioritize which are based on the criteria, the economic importance in India, an immediate need for building for trait specific root stock is very important, and level of threat, species, and most vulnerable area, conservation requirement for posterity, gene collecting, the region ecology context. And we need, while prioritizing and for finalizing, we need to keep the mind the what are the resources available. So we have to plan it phase wise, maybe 5 or 10 years like that. Towards maximum explorations we can conduct. Yeah, that we have to keep in mind. So this is another way the prioritization based on the gaps, because significant amount I have already shown you has been already collected in the gene bank. So more than 450,000 accessions are already in the gene bank. ICP. I should leave the pointer. So here in this case, if you look, if you see that there were more than 99,000 accessions the rice or gene bank, but we had the location details of only 55,000. There were 54,000 nearly, which for which the site were known, not known. Actually, what happened earlier, there was less number of states, 22 like that, 28, now 30 like that going on. And number of districts also have gone around 70, 680 like that. So earlier and many material we got from the different sources also gave us the correct information of the area of the collection. So in National Gene Bank, there are Ankur, can you come? These red dots are going on increasing on the presentation. So why it is going? Why they are going increasing? Please. So this is a gap we have Right. Share.
Sir, please unshare your screen and then share another.
Share again.
Again share. Sir.
Now all right, na?
Yes, sir. Please unshare, sir.
No, now it is okay.
Yes, sir. Now it is okay, sir.
Okay, okay. Mouse नहीं use करूँगा ना? Mouse.
Sir, voice also some sir
Okay,
technical
okay.
glitch is coming, sir.
Okay, okay.
Yes.
Okay. So like this, whatever the accessions has been collected in case of rice 45,000, these are the notes. For yes, Iqbal will give you more details on these. These notes that the one that to longitude location of the country. So you can see the how much collections, which area we have already collected. Here for the wheat also, the maize, foxtail. In the previous table, previous table. 22, human agriculture, we have already analyzed gap in such these details. These like this map we prepare, and based on that that's why I was emphasizing on please do not conduct exploration independently without involving the DPGR, without our knowledge, without checking the data. Because if it is a collected, there's wastage of resources, and in the gene bank we will not keep the. So this is another map with respect to the wild 16 wild species of the Solanum in India. The number of species are given over here. Where their distribution is also given over here. Like you can see these are the areas where these species has been collected. In the entire state-wise, all minors and access. This way we are analyzing, planning, then finding gap, and we are accordingly scientifically proper process we are following conducting this. This is the about the apps database. This will explain you in detail. So this database we are using for conservation database, collection database. This will give you more detail of this. So coming to the exploration, first planning, we must acquire knowledge of the status of the species and area. So ecogeographic data must be gathered, and that helps to identify probable site for collection, distribution, range, taxa, etc. With respect to the land races, I would like to say are the major focus, only item which we have to collect, because we are not going for collection for the released variety. We are going for the traditional farmer land race. And in many of the majority of the cases earlier, they were in cultivation, but now they have gone from that area, have been replaced by the high yielding hybrids or high yielding varieties. If we go for any for land races, the Haryana, Punjab, Western UP, for the rice and the wheat, there is no land race, not available. So while going a particular area, we must have knowledge whether this any material for which we are going available or not. Otherwise, that will be a wastage of resources. For that purpose, for the biorelatives and other we have to gather information about the vegetation, potent species richness, then the seed maturity time, etc. That we can gather through the floristic and botanical literature, like the visiting local and national area, like our scientists go in Calcutta. Consult earlier exploration reports. And we must have the knowledge of the biological characteristics, like the breeding system, ploidy level, pollen efficiency, stem size, age, like these, whether it's set bird pollinated, they have more genetic diversity as compared to self-pollinated. Then we have to decide that. So we have to decide the type of survey. There are two types of survey we conduct. First is the coarse grid survey, mostly in unexplored area, and we cover the larger area, capture overall diversity. Grid survey is to build up the more collections where already some collections have been made. We want to get the information the specific trade from non-identified populations, previously explored area. The visits, as I have told you, certain districts and collections we have to go for the visits also. there could not be considered, yeah, or poor ecological coverage is there. It is by the But important thing is for going to the National Park Wildlife Biosphere Reserve protected area, we have to take the permission from the CCF Wildlife. That is the First Conservation Act. That is essential. We cannot enter into this protected area, National Park Wildlife without having prior permission. First, very important. That's a major PHB. Coming to the explorer, has to have sound knowledge of plant genetics, geography, systematics, botany, ecology, etc. Important is he or she should be a very physically fit person. Going in higher, difficult remote and tribal areas and of sound health, capable of working in difficult situations and withstand climate adversities. Good driver, be a good driver during need.
है ये नहीं मालूम तो वो देख लो अंदाज से लिख दो बस तो वही जो
Please.
स्प्रे करा ना वही डेट लिख दो
So,
Please unmute. Please mute your speak. Yes.
While planning, coming to the exact planning phase, one should not control. Here, here.
Sir, continue, sir.
Okay, okay.
Yes, sir. Continue. Yes.
So, as I have shown you, after making a going prior to the explorations, he or she should make a itinerary of targeted area, liaison with the collaborator well in advance, day-wise itinerary. Send the proposal well in advance, date of the person, and that is he or she should must deposit the seed of collective material in the gene bank, field gene bank at the earliest, mandated to assign IC. IC numbers also I have to explain, which is covered activity by this division. So guidelines for exploration activities, the scientists or whoever the team must establish contact with the grassroots level personnel, local institutions, reserve accommodation well in advance, remind before the depart, and identify the collaborator, which should be a breeder, having knowledge of the exemplar, working on that system. We do not allow more than 3 persons in a team of explorations because in the vehicle there are only 5 persons in the vehicle. One is driver already with them. Then 4 seats are remaining, so there should be one seat vacant to take any help for any person in the area, either farmer, your local help. So don't send any more than 3 members team. Next. 2 to 3 weeks mission. First camp, explore surrounding area, move to next camp, and like that. So these are the items already given what we need while going on explorations. I have been already given in the write-up, which you can see over there. In that. Then after reaching point, you must have meeting with your local person, those who will be collaborating with you. Fine-tune the route, etc. Also survey the local market also. Fairs, district level, block level competitions, that gives you a good idea what are the diversity available in that area. So that is also important. This it's very important. The diversity. Go to the market, vegetable market and fruit market also, that gives you an idea what are the available in the market in that area, because the local material comes to that. Then coming to the collecting PGR, what forms the species level natural introgression is very important. Species, that's very important. So arbor varieties and that's main material which we collect. Variants in old varieties, source of adapted genotypes in varieties, wild BD forms that we can collect from the farmer fields, orchards, then home garden, backyard, wild habitats, forest, wasteland, etc., botanical gardens, like that particular genetic stored within the This is also the survey. We have collect, you have to go and visit the areas. Then what to collect? You have to collect the seeds, fruits, vegetables, oils, in vitro meristem, pollen, herbarium, salsa, herbariums. For genetic diversity augmentation, collection in the form of seeds very hard. In breeding elite trees, unique traits. Means the clones they are prepared for that genotype, prototype material, then science, so but is collected. In case of recalcitrant desiccation, told us tolerant species cannot tolerate desiccation, drying of that. The fruits are collected. Genetic diversity samples populations of are collected. Then when to collect, this is important. Is to collect the physiological maturity 5 days before crop. And for crops also. And for standing crop, annual crops also, it's better to collect at the time of the maturity in the going to the crop so that we can do proper sampling. 2 times identification per 2 times visit first, identification marking the tree, then going for collection. Then for orange season is best for herbarium. Important is avoid days of festivals and elections. From where to collect the priority should be on fragile ecosystems like coastal areas, solar, swampy areas, cold arid tracts for adapted traits, and the transition zone between 2 subspecies. There are lot of morphological variation is found. That should also be taken into latitudinal longitudinal variation. Brings out gives good diversity in protein qualities and ingredients like that. Breeding images use old chart, uniform age, elite trees, hotspots for particular sap as shall be looked in for the system material. Abiotic stress like the salinity and the heat, like Rajasthan. Yeah, from where to start? I start from the prior tracts versus prior tracts. The crop matures first as compared to the valleys and goes up to the hills. If you see in the case of mango, it starts ripening first in South India, then it comes to the northern India. Like that, we have to move. Then going for exploration, making your trip. So what you can do, you can make a route as shown in this picture in the red round color. And but certain areas like the Son Padum Son Door, where there is no other road come out, you have to go Janskar Valley, come back from the same. Better to collect cover the maximum areas. Then going for collecting, prior to collecting, check fruits and the seeds, whether they are not empty or immature seeds. In case of citrus, many species get to very less number of seeds. So it is the major germ plant, which we have to germ cells, germ material, productive material. We have to consider if that is not available, so that your activity for waste. Plastic food, there are certain bags in which you have to collect the plastic bag, plastic fruits, vegetative produce, most you require, and on seeds, paper bags like that. You have to collect, go with the material. And how much to collect? 2,000 seeds for self-pollinated crops and 4,000 seeds for cross-pollinated crops are required to submit to the gene bank. That's very essential. Otherwise, the samples are rejected by the gene bank. So, besides that, you need to collect for the characteristic purpose. Listen to the 2004. Then fruit species collections differ from other crop species because they are perennial trees. Then they are some of them are under domestication stage, hence best available farmers field and in the wild. Majority are out developed specific variations so accordingly sampling and frequency. Not flower every year. Periodicity there's some say in mango. Every year do not produce fruits. Ratio of fruit to seed mass is often very high, but that requires different handling process. Mostly recalcitrant seeds fruits. We see the specific protocols required. That's the limitation for us. So it is our priority nowadays. Then require collect vegetative material, immediately transport. Fruits are generally at the height, so that is but how much number of root stocks or the samples we have to collect. The number of fruit stocks are 10 per sample, then at least 8 grafts may survive. So 10 must be. So that means root are 15 to 20 numbers, so 8 to 10 each, that would be sufficient. These are the things which we have to take care of the material. So rooted suckers, fruit cutting, etc. There are several things which we have to collect. Then there are hardwood, softwood, that we have to deal with. So we various recalcitrant. Seed, we have to go for the seedlings. And we have to look at the viability also. So these are the things. And coming to the sampling method, we have to capture minimum 0.5 greater than 0.5. The all alleles 90%, so that capture 95% allele minimum. The sample. Random sampling, generally we go for that, collecting single spike, panicle, fruit, berry, for at least 50 plants from a number of transect, like over here, zigzag method. These are the sampling things. We are also shown in the picture that the team is going in different places. So there are different collecting techniques also required, particularly for fruit trees and seeds also. Don't go for the fallen fruits because we don't know from which tree it has fallen. So it is a very cheap with better suited for collecting, but uncertainty of contamination, poor quality. Then removing fruits from canopy using ground access by the different methods. Prior to collecting seed flowers, check number of seeds mature. While collecting, recording information on traits, quality, suitability, etc. And meaning the name of land is very important to ask to the farmer what is the meaning, because almost all the land they have a name that must be recorded. They are collecting equipments which we must carry, secret we must carry, knife we must carry, not like this. How we are collecting that. But climbing on the tree should be a last resort. So we should not go. It is a risky. If anyone person fallen, and then we have to go for the compensation. Do not go enter. You can carry the root stock also. The vehicle itself. On the side, it can be transplanted. Branch grafting can be done. Another important. You can see in the first left side picture, these are the their roots. Seedlings are being carried, then it's grafted. Transportation of the seed bank or your institute takes several days. It's advised to dry the seeds, take a gel to carry with us, dry in charcoal in sealed plastic bags. We must frequently see every day the seeds better shade dry in the staying place itself, provide halting point in the same leaves to be collected for the DNA extraction also. And for the previous identity also happens in many cases. We don't know the name of the species. Very important is that to collect the herbarium and take the photographs for authentication from the list. That's very important. Points to remember is that explain the when we are contacting the farmers, we must explain the purpose of visit very carefully, approach them tact, behave politely, very informal, not so that we are so highly educated scientists and all those things. We are one of them, like that. And age people are the best source of information. This custodian farmer, like in the right side the picture, you see Prahlad Sahu from the custodian farmer currently having 267 rice plantations and growing them. If you see in the picture, like the scientists, we grow all maintaining all records, etc. These are the first like farmers we have to. So it is a art of extracting material from the farmers. The old ladies, they generally do not share the material. We have to get them very. So many times you find only ladies in the house. So take any boy or some person, don't go alone. Important thing is drivers should not be involved in the task of locating. We left to drive only. And take SUV like Bolero. and go with appropriate clothing, gloves, etc. Umbrella, etc. Trusted areas, habitats, leech, etc. Low sun, mosquito, these things carry. Always keep a route map, GPS battery, target area before entering to the orchard, contact to the farmer, call to the farmer. First, take the help of forest guard. Always inform to the local authorities. Have important telephone numbers. Keep your identity card, letter from head of the utilization before going on the business. Honor social customs. Focus in evidence of target. Talking, discussing with ladies, be polite. Don't talk at their religious and political matters. And keep a flexibility really. If there is a weather conditions with rainy season or something like that, the festival comes so that you can increase or decrease like that. And personal take into account the personal safety. Report what you do. In the forest, there is a guard is available. So handling, daily inspect the material, see, dry the room before going to sleep. Slightly underripe fruits kind of unnecessary in the collected. Entire fruit or the bunch also. That branch allow mature and well ventilated. Transportation bags takes several days, so advisable to dry pack and send back to the station. Frequently inspect the material, daily inspect the material. So in the halting place, like the herbarium is being made, like the this is the newspaper being used for drying and cleaning process. Then this is a field passport data book which we carry, important informations which we have to enter, all given over here. So this date, collector number, botanical name, these are the very important things which we have to record here. So in case of the perennial fruit trees species where it's not possible to grow like the annuals, so in situ characterization during exploration is also feasible. To record back the number of characteristics, site itself, depending upon the descriptor. The post coming to post collection handling, seed extraction, packaging, labeling, etc. This we have to do report writing, etc. And then it has to be sent to derivation division for allocation. And with share with the collaborator for multiplication and for correction. Report writing is very important. There are certain points which you have to write in the report. It's not possible to get. This is important. Passport data sheet. This exploration division is providing the IC number, dealing all the material across the country comes to the exploration division, higher exploration, for which this is given here. The downloads, NBPGR in download CID 112. This is a passport data sheet. You have to fill up that. Collector number is very important, which is the name of the collector, means the abbreviated first number, first name like that. You have to give that. So all the things which already given or in that tips are given in the on the website how to fill this. Important is the biological status. Fill in that. Wild all species other than cultivated, crop wild date is belonging to gene pool 1 and gene pool 2. Gene pool 1 and 2 we mostly we collect because there are possibilities they can hybridize, cross, and use them. Okay. And VD form of cultivated species, companionship in the field, like land race, traditional breeding line, light line. The scientists face difficulty to what give the biological status. However, this AKMU, they also stick too much. So this was all. I think, but 5 minutes late. So thank you very much. This.
Sir, thank you, sir. Thank you very much for your.
Any
valuable
questions?
Yes, sir. And please, all participants are requested if you have any doubts or you want to interact regarding collection methods and procedures, you can ask.
I mean, see, it was a long lecture and I have to go fast. Everything cannot be explained, but it's already given in the writer. there are publications. So first is the IC number. What is this exploration division you have to deal with? Want to collect explore, then you have to send that well in advance. This is the plan we are making. We are carrying out always all the explorations involving the collaborators, involving the strategic. The institutes, but many of the scientists are involved in the development of the varieties. Work, they generally not company, they will not like to join because they also have that at the same time their crop is grown, harvesting. You understand? Any questions?
Ask any questions.
Anything you would like to know regarding exploration?
Sir, one of the participant asked, sir, Dr. Vishnu Gati, what is biological status of field crops?
Field crops, they are cultivated. They comes under cultivated or material in the institute. There's a breeding line and breed line, research line like that. Important thing is in case of the breeding material, they have to give the latitude, longitude of their institute. And in another thing, while giving this latitude, longitude, give this in decimal format instead of degree, minute, second. Because nowadays, this database is taking, you can convert it. Conversion is available on the Google and in the app also. If you choose that part, so you can give it in the decimal format.
Yes, sir. Sir, Dr. Malarviji, she asked how many seeds are, that means for tree species, how many seeds to be collected?
How many seeds to be collected from the tree
For
species?
tree species. Yes, sir.
See, the thumb rule is 2000 for self-pollinated. But it depends on the for tree species. Generally, we should go. There are two types of collections for trees. First is the gene pool collecting. In case of the gene pool collecting, we take the entire diversity available in that particular area. So this can be several number of tree species. But for tree species, generally, it's not possible to grow them, evaluate them. So better go for the elite, the best tree, plus tree, which we call. Okay, collect the material of that one, and it's better to collect only the seed of that plus tree. If it is a cross-pollinated, then go for the sign wood, bud wood, or vertical, or the gene. And in case of the field gene bank, maximum number of trees per accession are grown. So we collect 10 grafts. So we presume that at least 7-8 grafts will survive, and only 3 will be planted in the field gene bank. So that way, for tree species, best is to go for the elite trees. And for seed, that's a gene pool that captures the entire amount of the diversity sample of the tree. Maximum, say, nearby maximum number of trees samples. That is kept in the gene bank. That's only for purpose of the seed in the gene.
Hope Dr. Malarviji, you got information from sir. And sir, one of Dr. Tangavendan, he is asking how to getting IC number for mulberry species.
Mulberry, we are already working with the Agriculture Institute, somewhere it is. Our experiment was conducted last year in Arunachal Pradesh. Our scientists have also collected from the Jammu Kashmir area. So, question was how to get IC number. For that purpose, the seeds are available. We should submit the seed. In case of mulberry seeds are, we will conserve the seed. We send the seeds, and we will issue the IC.
And sir, Dr. Anurag requested, sir, your presentation is very informative. he wants, if it is possible, share the presentation like that. Sir is asking.
Yeah, we will share.
Yes. And one thing is we given write up in compendium also. That is also Sir given very exclusively, briefly he represented the collection procedures.
Yeah, it's almost everything is given over there in the right.
Yes, sir.
So if there is no questions, thank you very much for patiently listening. And I want to say that the exploration division conduct explorations needs your help everywhere. We go to the any area, we go to the universities, take their help, take your help. Because with that area is not known to us. Without your help, we cannot go, cannot do these activities. So we at NBPGR look with close collaboration, close help with you, all people, and when joining that lecture, this training program, you will gain the knowledge what we the NBPGR is doing, how we are carrying out. This was in very brief, but definitely you will understand the importance of this activity. And I understand that we don't need to explain after going over there that what for we have come and what was the basis, how we came to you for that particular area. Because 450,000 accessions already collected, already in GenePath. So now we go only for the based on the gaps where the material has been not been collected or underrepresented in the. There is a possibility of getting. So not more than 3 districts. If you are sending a proposal, not more than 3 districts we can cover in 12 days, 12 to 15 days explorations like that. Please take care of that. If only one crop, then it's possible to cover more number of days. Only for one crop. Otherwise, you can join it. You can send your demands. We are preparing the exploration plan in the last stage. So if anyone want to go on exploration, want to collect particular material, you can send a demand to have exploration. Okay, thank you very much for joining us.
And sir, dear participants, for your kind information, we already started the collection of ornamental germplasm also. Last year also our regional station Ranchi, they conducted exploration exclusively indigenous ornamental wild species. And these are very important germplasm. Now we initiated the ornamental germplasm also for collection all the procedures. And anyone is having any doubt, please you can ask. Okay, I hope all the participants and currently next sessions will come and you will get information and remaining all other information of PGR aspects. And next we are taking a small break for tea and we will join at 11 o'clock. 11 o'clock National Herbarium of Cultivated Plants. One repository lecture is there by Dr. K.C. Bhatt. And please join around 10:55 like that. Please, sir. Thank you, sir. Thank you very much. It is.
I went very fast because considering the time given to me.
Yes, sir. Yes, sir. And sir, yesterday, sir, lot of people asking regarding collection of germplasm and these things. Sir, and sir, Dr. Taraknath Saha, actually, he is a renowned person, sir, in floriculture. He is a senior scientist, and he asked, sir, we want to participate in germplasm exploration.
Dr. TN Saha, DFR.
Yes, sir, DFR. Yes, I am. Yes, sir.
You want to join? Or?
Yes, sir. We would like to join for exploration process. Actually, this year our scientist Dr. Ganesh Kadam has joined the exploration in rose cultivation in Uttaranchal. So beside this thing, there are a lot many native ornamentals are there in our country, like in Eastern India as well as in the Western Ghat area. So in future, if you are having any plan in this, we would like to join from our DFR side, sir.
Yeah, that's I told you that we are already in the planning phase.
Right.
And so but you need to identify the particular area, the species target area.
Right.
You identify that area and we are going for exploration particular in that area. So we can definitely take you not more than one scientist. I already told you not more than 3 persons in a team.
No issue, sir. No issue.
Only one scientist. We can
No issue, sir. That is not a problem. Our
Because
director, sir,
we-
Dr. K.V. Prasad,
we-
sir, is very much interested in all those things because it is getting importance nowadays due to many properties like essential oil, these things are also there. We are also in DFR having more than 130 varieties of chrysanthemum. And we are evaluating all this for not only agronomic traits, but other like essential oil, other phytochemicals. So we want to enrich our germplasm of chrysanthemum also. So that is our intention.
Yeah, Dr. Ravi will share with you the exploration plan.
Yes, sir.
Okay,
Yes, sir. Sure, sir.
whatever the best you find, you can join
Yes, sir.
that area. But let me tell you, almost all the institutes are facing their financial problems. This year, we thought to have persons more in the northeastern region. Though every year we are conducting 6 to 8 experts in the northeastern region. This year it will be more. That the funds available in the northeastern NEH scheme can be utilized.
Right.
This year we faced a lot of problems. Several plant experts shortage. In the northeastern region, if you would like to come there.
Yes, sir. Not a problem. Sir.
Yeah, you can come.
Yes, sir.
Thank you, sir.
Okay, dear participants, please be in online, but we will join at 11 o'clock. Yes. Thank you. Yes, sir.
Thank you so
Thanks.
much.
No,
Right.
वॉइस इज
ओके सर। ओके।
मेरा ठीक है।
हाँ ठीक है सर, वॉइस क्लैरिटी आ रहा है। हाँ
ओके, वॉइस क्लैरिटी है ना।
सर एक बार आप शेयर करिए सर पीपीटी अपना।
ओके ओके ओके, एक मिनट है।
सर मैं आ रहा हूँ।
कहाँ शेयर होता है यार, मुझे नहीं पता।
तो शेयर स्क्रीन में जाइए सर। जस्ट फॉर
हाँ ये
प्रिंसिपल साइंटिस्ट and head of the division of plant exploration and germplasm collection in NBPG or New Delhi, and he is a suitable person for exploration for collection of agro horticulture crops. He conducted more than 60 explorations as a team leader and collected nearly 4,000 accessions from different parts of agro biodiversity hotspots. And currently he is a curator of National Herbarium of Cultivated Plants, involved in management of herbarium activities, especially of cultivated plants. And he reported a new species of 4 new species and new records or uses or ITK aspects. Nearly more than 150 ITK aspects are there, and genetic stock registered one genetic stock registered and fellow of ISPGR and review of IJPGR and other journals. And he is facilitated as ethnobotanist by the Society of Ethnobotany, and his vast experience in germplasm collection and ethnobotanical studies and medicinal botany also. And his publications are more than 55 publications and books and bulletins. He is having and some popular articles also there. And he is currently working in the aspects of collection and exploration mostly. And now he will deliver a lecture on National Herbarium of Cultivated Plants. It is a repository, national repository. And sir, I am inviting you, please kindly deliver your words on a National Herbarium of Cultivated Plants, and please explain and how to deposit the specimens and methodology methods. And please sir, please continue.
Okay, thank you Dr. Ravi. Am I audible?
Yes sir.
Okay, so shall I start my talk?
Yes sir, yes sir. Kindly
yeah.
Yes.
Now it is visible. Hello.
Yes, sir.
It is visible.
Yes,
sir, visible sir, please continue.
Yes, sir.
Okay. So my today's talk is National Herbarium of Cultivated Plants, that is NSCP. It is a national repository at NBPGR. So I will just share my experiences and what is preserved in this NSCP that I will discuss here, and how to prepare herbarium, I will also discuss about the preparation and collection of herbarium and what other studies we are conducting in this important repository that I will discuss here. So, first of all, we should know that what is a herbarium. In previous lecture, I think you might have just heard about the exploration activity. So in that activity, that identification is very important part. Unless you identify any plant, any species, any germplasm, it has no use. You cannot preserve, you cannot conserve. So its identification is very important. So this herbarium, what is said that whenever you are going for exploration, we should visit herbaria in your area, either a university herbaria, traditional university herbaria, or BSI Botanical Survey of India. The herbarium are available in different parts of the country. So before proceeding for any exploration, you should visit the herbaria, and that herbarium will help you in identification of any new species, any collection what you are going to plan. That it will help. It is a very great. It will help you in a lot. So your visit to the any herbarium is very important. That you know that it is this herbarium is a data source of for research. Basically, as I told you, that identification, it is based on taxonomy and what is the distribution of plant groups. And it is also help in the writing a flora. That flora is very important tool for identification, for knowing about the species in particular area. So the floras are published by many taxonomists for this identification for the vegetation, the diversity available in a particular area. So this is one of the important tool, and it is also an historical record for plant locations, that it will help you to know the where it is locality, where it is located, this particular plant, particular germplasm, so that you can easily visit that area for collecting the material. And it is a repository for voucher specimen. So whatever you collect that in the form of herbarium, it is a repository for voucher specimen related to published scientific reports, and it is an educational resource. That definitely it is an educational source for learning to recognize the plant species of an area, what plant species occurring in a particular area. It is an educational source. Anybody, students, researchers, they can visit the herbaria and use this herbarium for as a learning resource. And it is accurate and permanent documentation. It is accurate and permanent documentation of botanical information. Whatever botanical information you want, anybody wants, it is available in that herbarium. So it is a very important resource. And first of all, what is herbarium? Its plural is herbaria, and it consists of a collection of usually dried plant specimen. Actually, the herbarium is a dried plant specimen. It is not a live. It is not a whatever new plant. It is a dried plant specimen, and it is arranged according to a accepted classification of system. So if anybody knows about the taxonomy, so definitely I am sure that he would know about the classification system. So whatever classification systems are mentioned, these plants are arranged according to the classification system. And the Bentham and Hooker, a system of classification, is an important classification system which is helpful, still being followed. There are so many new classifications are there, but this Bentham and Hooker system is followed still. And in 1700, Turnford first used the term herbarium. Who used this? Turnford first used this herbarium to describe a collection of dried plants. He described the herbarium. It is a collection of dried plants. He described this herbarium term, and how it was originated. This is a Latin term, herba. Herba means grasses, pastures, and it is derived from the Greek, which is in turn appears derived from Sanskrit, bhar. It is a Latin dictionary in 1879, and the areum refers to a place where you are keeping them. It is the definition of this of herbarium. So herbaria are centers of research. As I told you, it is a center of research and learning about the identification, nomenclature, classification, and distribution and use. Okay, this is some this past history, that journey, how herbarium was started, prepared, how the people they started the preparation of herbarium. So that the earliest herbaria was a physic garden, because the earlier physicians, they grew medicinal plants, and they prepared for their future as a reference sample, and this was also used to their students for as a pharmacy students or physicians, so that they can learn how it looks and what is the name of that particular plant. That for that purpose, earlier the physicians, they used to prepare herbarium for learning. And this first in Luca Guinea was during 1490 to 1556, an Italian physician and botanist created the first herbarium. He was the first creator of the herbarium, Hortus Siccus, and oldest surviving herbarium dated back to 1532 by Gerardo Chivo, a herbalist and student of, he was the student of Guinea, Luca Guinea. So this is the sum of the past history of the herbaria. And then what you must know, what is the oldest herbarium in India? That is the Cyprus procerus. It's a grass. It is the oldest herbarium, and it is available in our BSA Calcutta. Then what is the significance of herbarium? You know, the herbarium is a comprehensive data bank, and it represents the plant diversity of a region and a useful tool to study a wide range of collections. It is, as I told you, it's aid in identification of plant specimens, and herbarium specimens can also be used in assessment of the risk. It can be important source for planning field trips. Yes, as I told you, it can help you in planning the field trips, where to go, which plant is to be collected, and its identification. It will help you in knowing the identification, this particular botanical identification. Herbaria with dried pressed plant specimens and their associated field data, essentially collections, photographs, also will help you in identification in any plant. Herbarium are also used for monitoring the climate change. If you will study the herbarium, that how it looks, if you visit the herbaria and whether it is available in that area or not, it will help you in this monitoring the climate change in that particular area. And it also provides the comprehensive information which is essential for studying, that it is a very important source for studying the taxonomy, as I told you, anatomy, systematics, ecology, morphology, conservation biology, biodiversity, ethnobotany, and paleobotany. So this is very important. What is the significance of herbarium? It's a very good source for studying many things. Even DNA you can extract from the dried plant parts, and you can study many things from that, as I told you, as I mentioned here. So it is in world, there is a catalog of published herbaria. It is known as Index Herbarium, and our NSCP is also mentioned that Index Herbarium is also cataloged in that. And globally, there are 3,001 recognized active herbaria with a holding of 38 crores herbarium specimens, and India represents over 3.5 million herbarium specimens, over 23,000 type specimens. This is the type specimens, new plants identified. It is about that. And it is a word catalog of herbaria. It is mentioned here. You can see. Then in the world, how many types of herbaria are there? There are major national herbaria. And what are the activities in those herbaria? That world and regional flora, it represents, it helps in research and identification, variation with respect to geographic area. Like in regional herbaria are there, research and identification of taxa of area and adjacent regions. It is one of the important activity in regional herbaria. Local, you know that it's helpful in, it helps in identification mainly, and teaching, mass gathering, and watchers of studies, photographic records is available in that particular local herbaria. And virtual, you know that nowadays we need a virtual mode, that so that we can carry it in the field, and for quick identification we can use this virtual herbarium. Then some students for their academic purposes, they also submit the herbaria, their herbarium in the national or in the regional herbaria. So the students also used to prepare herbarium and submit their herbarium in the local herbarium or national herbarium. So some important herbaria, you must know, we in the world, how many, there are many herbaria are there. So in France, that is Museum National d'Histoire Naturelle, having 8.9 specimens. And then there are so many herbaria, it is mentioned here. In the lowest herbarium specimens are available in the Beijing, that is Chinese National Herbarium. The 2.47 million herbarium are available are preserved in these different herbaria. You can see here the number I have given here, and the name of herbarium, and the location is mentioned here. Then in India also we have got a number of herbaria. Then, and you know that the Central National Herbarium at Calcutta, acronym is given in there for this, for reference. If any herbarium is consulted and you are mentioned in any publication, you can mention the herbarium, the particular plant, and then with this acronym CAL, it is localized at Howrah, in Kolkata, and how many number of specimens, more than 20 lakhs specimens are preserved. And this is the biggest herbarium in the India. That and then there are other herbaria or forest research institutes at Dehradun. We have got good herbarium. You can consult, you can visit. And the regional centers of the Botanical Survey of India, in every part of India, we have got in Assam, Sikkim, and many other, Dehradun, many other places, Pune. So you can visit these are the herbaria in India, for Botanical Survey of India, FRI, Bladder Herbarium at Mumbai, and National Herbarium of Cultivated Plants that in our in New Delhi, NSCP. This is only the different herbaria. Then this the herbarium it mentioned here. This is somewhat different. I will just explain how it is different. Then it is said that that I just mentioned that these are the dried plant parts. So herbarium are dried plant parts, and it is mentioned that dry garden need more care. These are called as dry gardens, and we need to take more care, because India is one of the 17 megabiodiversity countries with over 11% of world's plant species, and herbaria across the country stored millions of rich biological samples, helping scientists to connect with the past and better understand evolution, extinction, climate change, and even introduction of invasive species. But due to inadequate funds, that this is maintenance of the herbarium is difficult. Without funds, you cannot maintain. So, but due to inadequate funds and little government support, these repository of the knowledge remain forgotten in cupboards. Okay, so the Biological Diversity Act of 2002 has further restricted access to these resources. So you can see, then Viva Sauni analyzes the state of Indian herbaria and suggests ways to increase scientific collaboration while safeguarding the biodiversity. So we should take care of the very good resource in our different herbaria available. It can help you in many studies. So that is appeal to you all, and also we need to take care of these herbaria. Then general advisory, how to proceed for collection of the herbaria. That in previous lecture you must have heard about that. There are some planning. You should plan that where to go, how to go, and when to go, and what is to be collected. Like these things is, again, you have this collection of the herbarium is also followed. It is an arduous job. This is not easy job. It is difficult. One must be sound mental and physical health when going to collection trips. So the weak person cannot visit, because he has to visit difficult areas, different areas, and for that purpose you should be sound, healthy, this mentally and physically you should be sound for visiting different difficult areas. So be clear about what is the purpose of collection. You should be clear why you are collecting. What is the purpose of collecting the herbarium? For identification, for academic use, for this diversity analysis, or for any other purposes. You should be clear that in your view why you are collecting, and make yourself well acquainted with the area where you are going to collect the material. Make extensive notes that whatever you have to get some information about that area where you are going to visit, on its boundaries, topography, terrain, forest types and cover, weather conditions, endangered species available, road network, rest houses available. And it is said that a casual approach is likely to ruin entire trip, leading to loss of time and money. So you have to plan well, and you have to gather the basic information about the area, about the crop, about the plant, that where you are going for collection. You should be clear about your purpose, should be clear, and you should gather information on these aspects. And if planning to work in a forest, as Dr. Alawat also mentioned, that if you visit the national protected areas, so you need a permission. That permission is very important. Prior permission is very important from Forest Department, because they will not allow to enter in the protected areas, restricted areas. So you have to get permission before going for collection. And always carry your identity card, because anybody can ask about your identity from where you are visiting. Anybody can catch you. Forest Department and any other policeman or some can catch you, that why you are collecting this material, why you are uprooting this material, why you are this plucking this flower and like these things, fruits and other any item. So that you need to require to have with identity card is very important for your identity. And if available, obtain the flora of that I just explained, that flora of any particular area, to know about the species, to know about the plants is very important. During exploration, you should carry with you a regional flora, or some published articles, or published flora of any region of that region, so that you can know that these are the species available in that area, and you can collect the plants of your interest. Draw up a checklist of the articles needed. You should prepare a checklist that what is the instrument you require during collecting the herbarium. Field book is an important, yes. In case of this earlier lecture, you might have heard that field book is an important. Rather, I think the passport data is important, but field book is why it is important, because whatever it is not mentioned in the passport data book, you have to mention, you have to record in your field diary. That field diary updation is very important. You need to have a field diary with you, and you can record what is not mentioned in the passport data. It is advisable to make field book entries on the spot, whatever you have seen about the plant, about the diversity, about the species available, about the association, about the local condition, about the local uses of that particular plant. You need to record on the spot in the field book, and number be tagged to the specimen immediately. The number you have to be tagged, that specimen what you have collected, and everything should be noted in the diary. If time limits does not permit it, location, date, habit, soil type, and local names should be entered immediately. Yes, immediately use it. Otherwise, you can forget when you bring it at your that where you are staying. Then it is difficult to remember that what was the habit, soil type, and other things, local name and other things. So you should immediately record in your field book. Then remaining data you can record when you at your camp, where base camp. And under no circumstances, this important task should be postponed until next day, and data should be recorded. You have to record whatever you have seen, you have observed in your diary. You have to update this diary whenever you are in your base camp before updating your diary. Otherwise, because why I'm saying this diary is very important. Whenever you are writing a paper, it will help you in writing a paper. It will help you in writing a report of that particular area, particular species, particular your mission. So this is very good. It will help you in so that whenever we visit we go for the excursion. So I request to all my colleagues or the fellow that whatever you have seen during collecting the germ plant, you have to update this information in your diary, personal diary, in personal notebook. It is very important. It will help you in compiling many things. So it is very important that whatever you have seen, where you have visited, the name of plant, the name of place, name of person, and the material available, diversity available, vegetation available, soil type, and other things, and the tribal people or the local rural people or whosoever is residing that. So everything you have to mention what you have done on that day, you have to mention in your diary. Then it is now we are discussing here about the National Herbarium of Cultivated Plant. That is a special depository for the herbarium holdings of cultivated plants. It is located at NBPR, nowhere in the world. It is one of the unique herbaria, that's National Herbarium of Cultivated Plants. Otherwise, in BSI, FRI, other traditional universities, they are having the herbaria of their own academic interests, plants, vegetation, and other, but we are having the herbarium of the important PGR value, having the PGR value that plants, species we are preserving. And this is among the 25 major Indian herbaria. Okay, it provides a reference collection. I know that everybody knows that this herbaria provides a reference collection of diversity in PGR. It is we are also preserving the indigenous and exotic material, whatever we are getting from the outside country, we grow it, and then we prepare herbarium for its as a reference sample for and preserving. Herbarium, especially, we are also having the seeds of the different plants where you are visiting. We are collecting the seeds also, as the NBPGR is collecting mostly seeds. So we are also maintaining the seed museum here. And photographs, yes, definitely is a very important part, that is photographs, taking photograph and making illustrations. These are very important parts of the herbaria. If you don't have the photograph, you don't have the illustration, in future you will not, you cannot refer that from where it was, how it was looking, how it looks, that you think these things you can miss. So that the photograph is very important. And if if you can do, then illustrations you can prepare of that material. Undertakes studies, and it will help in undertaking the studies on taxonomy, processing diversity, biosystematics of taxa of Indian origin or our native taxa. It will this source for the studying these type of things, taxonomy, processing diversity, biosystematics of taxa, prepares and provide aids for identification. That know that it is this provides aid for identification, field correction of crop groups. That we are mentioning here the as we deal the PGR. So we are conserving in the LTS that the crop group wise collection. Here we are also mentioning that how many accessions, how many herbariums are available in different crop groups. It is mentioned, it is available, preserved in our national herbarium cultivated plants. Identification services, definitely we are also providing identification services and issuing the authentication certificate for research purpose, for referral purposes. We are issuing authentication certificate. That we are why we are. I am saying it. It is we have also already started this for the pharma. These students and other students, they can also help. And for that we have already mentioned the guidelines in our website. Anybody can consult for how to prepare herbarium and how to apply for the authentication certificate. Establishes, and we also maintain a liaison with other herbaria, the gene banks, knowledge exchange and information. And we actively undertakes teaching in field of taxonomy and herbarium techniques for crop taxa. And this is the specific feature, or these are some different specific features of the national herbarium cultivated plants. It's how it was started, that this started from a small room at this IRA, and you can see our one of the senior colleague, Dr. Rosnair, and our this one of the famous botanist, famous explorer, Dr. R.K. Arora, late Dr. R.K. Arora. He has contributed a lot in the field of plant genetic resources, and he initiated this facility at NBPGR. And before that, it was at NBRI, and Dr. Chang TT Chang is here and it was started in a room measuring 5 into 5 meter in old campus of ICR and BPGR, where I am sitting. Herbarium specimens and seed economic seeds of economic important plants were preserved. And it is mentioned that when it was started, the Herbarium of Plant Introduction Division of Indian Agriculture Research Institute was reportedly set up in 1948 at IRI. And then there were more than 500 specimens of introduced plants, as well as evaluated and breeding material was preserved during 1948 to 1974. Then it was shifted in 1985 in as a National Herbarium of Cultivated Plants at National Bureau of Plant Genetic Resources in 1985. Systematic explorations and collection of herbarium was started from 1985, and then currently it is a big facility for using in taxonomy, teaching, research, and documentation. Now this is the herbarium, and at present I am the curator of this herbarium, and we have other staff that just Dr. Ravi is there, our one of the senior colleague, Dr. K. Pradeep. It is at at Trichu Center. Then this Pankaj Kanodia recently joined this bureau, and then Rita Gupta, he is a technical officer here, and she is working hard to maintain our herbaria. So this is the picture of herbarium now, herbarium, and now it is now we are renovating this survey facility. Now, what is the holding of this National Herbarium of Cultivated Plants? You must know it is the acronym is the NSCP. Then we are having cultivars that cultivated plants, primitive types, land access, and crop varieties. These are very important source for breeding material. And VD types, we have exotic and also the minor economic species, as I just mentioned. And this was started with a project that establishment, build-up, and maintenance of herbarium and seed museum of cultivated plants in the form of the institutional project. And we have got about 4,360. The total specimens we have more than 25,000, and they belong to 4,364 taxa and 1,545 genera, and they of the 267 families. They belong 267 families, and we have got the type specimens that news material new plants collected of 47 and 27 by NBPGR, and the others by contributed by others. And then seeds also we are having that I just mentioned 3,153, and also economic products like suppose we visit the area and where some specific material is prepared by local people, we bring it here. Also some specific material, if we see that in the field, we collect them and preserve here as a economic product, and these are 745. So this is the National Herbarium of Cultivated Plants. You can see the cabinets, and we are also having the facility for digitization of the herbarium. It is that we have it, so you can see the scanner is there, and we have the capacity of more than 50,000 specimens we can preserve in our National Herbarium of Cultivated Plants. So this is the oldest specimen in the NSCP. We have got the oldest specimen that you can see, Udania cogolens. It's a very important material nowadays. Very few, it is a rare or rarely occurred occurrence is rare. So, and Amaranthus paniculatus also, that you can see in 1932, the Vidane congress was collected, and details about this, it is mentioned in the, and about the Amaranthus paniculatus, again, it was collected in 1932. So these are the oldest herbaria, herbarium at our NSCP. And what is the procedure? What is the procedure to collect and to preserve, from collection to preservation or the storage? That these are some of the steps. We collect, go for the collecting the herbarium. We that is the procedure, that we press it in the field, then dry, mounting, identification, and storage, and then digitization, and then use. We are having the web, this digital information herbarium also. So what is the planning? Wow, how to plan? Planning of experiment. You know the weather condition of that area, duration of a trip, how many days you will spend there, and then how many people you will accompanying area, which area you are visiting, mode of conveyance, what is the mode of conveyance, the place of stay, where you will stay, purpose of collection, and other such factors as I just mentioned earlier. And what are the important this material you have to equipment you have to carry, that it is, you can see here the GPS. Nowadays we don't need the GPS, we are having in our this mobile phone, GPS facility is there, and this other camera is very important. And if in the field, in the laboratory, you need a microscope and Caesar, and this secateurs is required. These are some of the very basic equipments, and this is the list of the equipments you can carry with you. This these are the lists you can see here. I will not explain it much, but there are so many things we have to carry with you: vasculum, plant press, and then the binocular camera, cutter, blotting sheets, field book, and GPS, as I told you. These are some of the important polythene bags, press, plant press, as I told you, letterheads of your organization, so that you can identity card definitely should be there, and old newspapers, if it is blotters are not there. These are the some of the important material equipments you have to carry. Otherwise, that other things, it depends on the season and other these conditions. Then what is the procedure for collecting? That is very important. You have to collect, not that like this our basic 10th standard students, they just collect in the name of herbarium. Just they will collect a leaf and prepare as a herbarium in their class, that in their this teaching. It is like this. Not you should collect if you want to study the PGR. If you want to collect the this herbarium, you have to cut a twig, and it should be along with flowers or fruits, or preferably both. So this is without not a vegetative stage. You should have in that herbarium flowering twig. It should be a flowering twig or having fruits, or maybe both. In case of small terrestrial, suppose it's a herb you are collecting, then whole plant you can uproot, and it should be collected. You can uproot the whole plant along with the roots, along with the leaves, along with flower and its fruits, and you can prepare a herbarium, collect a herbarium. You can put in the vasculum. If the taxon is common and represented by a good number in a proportion, if a population is good, then you can collect 5 to 6 specimens. Otherwise, it is not recommended. 1 or 2 specimens are sufficient. So if it is population is good, you can collect 5 to 6 specimens. But if it is a location where you are having less, this material is not in good population, you can collect less than 5 specimens. Then the collection has to be avoided. Not more than 5 specimens is recommended, but I will recommend only 2 specimens you can collect. In that case, try to take good photographs. Yes, if it is less, population is not available much, you can take the photograph and record the characters in their field. Whatever the characters you can see, you can record. Without collecting a twig or whole plant, collect 1 or 2 flowers for study. If less population, in case of, you can collect one flower, or for the one or two flower for the study of that, because flower is an important part to to know, identify the plants, so that you can see the collecting, how we are collecting in the field. And as you can see here, immediately whenever you collect an herbarium, you have to press immediately in the field itself, because otherwise when you put in the vasculum or in any other form, you can, it will not, you cannot prepare a good herbarium. So immediately you press in the blotters or newspaper, whatever you are having. So this is a herbarium press, and the pressing is, yes, pressing is very important part. Pressing and drying, they are the two simultaneous process in herbarium preparation. If you will press properly, definitely, and press and dry, and if you exchange the pressing material, the material in which you are pressing the material, that you have to change blotters, or either it may be a newspaper, or it may be a blotter. So you can change in after 24 hours or every day. And before pressing, thinning of specimen is to be done. So not crowded specimen, you can, otherwise it will not look, no, you cannot study the characters, and you have to remove, or these part from, either it may be leaves or stems, or maybe branches, or infected portion. You have to just remove this. Leaves should not be crowned on the top of each other. Press the collected plant parts immediately after collection. As I just mentioned, bulky parts, if you are collecting the bulbs, bulky part, tubers, it may be reduced by slicing. You can slice it and press. Then some leaves should be turned upside. If you are, as I just mentioned here, that you sow some leaves upwards and dorsal, and some leaves as a ventral side, so that you can study the leaf is trichomes and other things, whatever you want to study about that leaf characters. Then hardy fruits may be kept for wet preservation, not for this pressing. As a dried specimen, you can preserve as a wet preservation in 4.4% formalin solution, or you can dry it separately. Then this is, as just mentioned, that crowded leaves that you can remove, and then you can see in the right side is correct method of this preparing the herbarium, that you have reduced the number of leaves, and this form, in this form, you can preserve, you can press the herbarium. And in case of this, as I just mentioned, tuber crops, you can slice it, press it, and from this metal presser, and you can prepare the herbarium of the tuberous crops or specimens in this form. Then the another step is mounting. That mounting, when you have brought it, dried material, pressing, as I told you, you have to change it after 24 hours, because it can lose its color, original color. So you just take care of that material. Every day you have to change it. You have to just dry it every day. And then now that I just mentioned, mounting is again a very important part that of this process of this herbarium. It is generally mounted in the 29 into 41 cm standard size of the the paper, heavy paper, and especially either pasted with glue. And when you are mounting, it should be glued or stitches on the mounting sheet. Gluing and pasting is best for flat leaves and small flowers. Yes, label is again very important. This labeling is very important because whatever you have recorded in the field, that label will mention that information about the species, about the area, locality, date of collection, other things. That is very important. After mounting, you have to label it, and you have to paste the right side of the plant, just as mentioned in this this herbarium. Label with all written typed information are generally applied glue and fixed on the lower right hand corner, as you can see, and the material can be removed from the specimen for further studies. And loose material like leaves, flowers, seeds, you can remove for other studies from the mounted part of the herbarium. Also, information, photography records, and voucher specimens of experimental studies can be made available on the specimen sheet. You can use this label and everything you have to mention in the label. Generally, what happened? The people, they just collect it, just they will write the name of the species and the area of collection, date of collection, collector name, but they will not mention the information about the area, about the use, about any specific feature of that species that observed in during your collecting. So that is very important part. Unless it has no such information, it is no use, because and in future you can publish information, you can record, you can use in the study publication, other things. That information, the very basic information needs to be recorded whenever you are collecting the herbarium specimens. Okay, so documentation and digitization. A referable identity number. Yes, whenever you are collecting, you have collected, you have mounted. Now, documentation and digitization is very important. You need to assign a HS number, identity number of that specimen is very important. If it is having all these things required for the herbarium, related to herbarium, you need to assign a HS number to that particular, every herbaria assign a HS number to that herbarium. So it is very important indexing documentation of that herbarium. For that purpose, you need to give the identity number, and a periodical method is followed to digitize the herbarium data for a record of further use. Yes, digitized, you have to periodically you follow the method to record for further use, and different fields are used. In case of label, herbarium number, as I told you, botanical name that is mentioned, local name, family name, locality, state, district, and this village. Notes, as I just mentioned, collected by, collected number, that needs to be mentioned in that label whenever you are indexing or mentioning the preserving the herbarium. It is mentioned here. All voucher expenses are digitized, that we have got the facility, and you can digitize for future this for using in future, or that digitized image is very important, and you can use the scanner for digitization of the particular specimen, and it's a safe specimen you need to treat, because the infection, the chance of infection is always there. So you need to treat by dipping an alcoholic solution in 2% of mercury chloride to protect from herbarium pest. So you need to treat it. You need to dip in an alcoholic solution of mercury chloride. Though mercury chloride is not recommended to use, because it is a poisonous material. So you need to dry it, or you can also deep freeze it at minus 20 degrees centigrade for 48 to 72 hours. If you are not using this mercury chloride, so you can deep freeze it, this herbarium specimens, so that no infection would be there. The treated specimens should be labeled. Once the specimens are glued, sewn, labeled, treated, poisoned, need to be indexed for filing. So now that final step is to filing the material. And then specimens, as I mentioned, it is filed, or it is arranged as per the recognized classification system, or as a Bentham Walker system. But at our NBPGR and NSCP, we are mentioning them in the arranging them as a crop group, and then crop group wise, and then alphabetic listing we are making of genus and species, and then type specimen, that if it is collected a new specimen, it should be kept separately in other racks from the main collection, and should be treated with much care, because this is very important. Specimen type specimens are very important, because they are collected for the first time and described for the first time, so that this you can see right side, that how these are placed in the herbarium cabinets. Now indexing, as I just mentioned, this HS number is to be allotted after mounting, labeling, and identification. It is unique number which gives idea of total holdings. That anytime you can mention in that unique HS number how many HS numbers are assigned. So it will give you the idea of the total holdings. Specimens in the herbarium are arranged as per a standard system, as mentioned. Type specimens are housed separately, as it is mentioned. Different colored genus folder for geographic regions are used to review a species general distribution quickly. Duplicate collection are appropriately numbered and placed separately and used in case of need for an exchange. And this is the format for indexing. The index card, serial number, herbarium number, species number, collection number, locality, collection period, and any other remarks. So this is very important things you have to follow during the indexing. Then yes, as I mentioned, that this is very important resource for establishing the taxonomic identity of any species. Here we have mentioned about the unknown species, that is a cucurbit. It was collected from the Nagaland, but it was it looks like a loofah, as I have collected it from Fake district of Nagaland, and as a loofah species I have collected. But later on, when we studied this material, and this identity was confirmed as a Herpetospermum operculatum. It was just mentioned in the literature, in the any herbaria, that only Herpetospermum was mentioned. But you can see its lower part, just you can remove it, and it's on that basis, on that character, we have made its species name as a operculatum. So in case of when herbarium species are not available, efforts should be made to raise plants to establish its correct identity. So in some time, if it is this, you collect the seed, then you can raise it, you can identify for its taxonomic identification. Characters which are lost, sometimes what most of the characters at the time of drying, at the time of pressing, at the time of carrying the herbarium, they can be lost. So you need to record like this flower color deflates, which may be said on drying, should be recorded. As I mentioned, field notebook is very important. In that field notebook, you can mention the lost characters, not drying, on the drying or other collecting. Now, sometimes the students, they visit the herbaria and our NSCP, they ask that how to preserve the lower plants like pteridophytes, ferns, bryophytes, and other lower plants. So there are some methods that ferns should be collected with the rhizome, because the shape of the rhizome and hairs and scales on the rhizome are important taxonomic characters. So it is somewhat different. And fronds without surai are of little value. So it should be like flowering plants, that like with the flower, surai is very important in case of pteridophytes, in case of fern. And for bryophytes, they grow on a variety of substratum. So when you are collecting, if you are collecting from on loose soil, can be picked up by hand. On rock or bark, collect with the help of a knife along with substrate. On decaying logs, sometimes grow in the decaying logs. So collect with the help of scissor. They collect especially in the sporophyte phase. This is the reproductive part. Phase for accurate identification. Without reproductive part, as in case of higher plants, in lower plants also the reproductive part should be there. It's a very important part for identification. If the liverwort is growing on a tree, collect some bark also. And specimens are collected in the paper bags and kept at cool and moist places, or air dried, preserved in dry conditions. Sometimes liquid preservation also used for thalloid liverworts, and specimens stored in herbarium packets made of brown paper. It is as per the guidelines of the Botanical Survey of India. Now, some research work can be conducted. Yes, very important resource for conducting this research work, like ethnobotanical information you can record from herbarium. It's about its use, about availability. Many things it's mentioned, as I mentioned, that in this level you have to mention about the area, about the use, about any other information about that. So it can be helpful. This herbarium can be helpful in the ethnobotanical information. You can see here, it's very herbaria is an underutilized source of ethnobotanical information. So you can record, you can report, you can mention that what information is recorded from different parts of it, which is lost because the ethnobotanical information is transmitted verbally, not through any records. So if anybody collects the herbarium, he can record the important information itself in the field. So some problems, some do's and don'ts are there whenever you are collect the herbarium. You have to take care. Herbarium is not a place meant for casual visits. So you have to just ask the curator or get permission from the curator, and only then you can visit the herbarium, because it's not a place for casual visit, because infections can occur whenever any person will visit the herbarium. So never forget that the large number of talented and dedicated workers have contributed to make a good and prestigious herbarium. Every specimen deposited there is a precious and irreplaceable. It cannot be replaced. Handle them with extreme care. So you have to handle them with extreme care. A herbarium is extremely vulnerable to insect infestation. So you have to take care of that. Infection should not be there. Okay, then always sanitize specimens thoroughly immediately before taking them to a herbarium. So these are some of the do's, don'ts, and do not write on a herbarium sheet except the textually important notation. So important, except the textual character, do not write anything. Do not remove any part from herbarium specimen without permission from the curator. Parts so removed should be studied carefully, then placed in an envelope which is pasted on the same herbarium sheet where from they were removed. Never misplace the herbarium seeds because they are arranged as per the some system. Take out only one folder at a time and mark its location with a card. Do not fold or otherwise mutilate herbarium seeds. So some of the do's and don'ts are there. So now, as I mentioned, the digital herbarium have been prepared by NBPGR and is available at our this website of NBPGR. The digital herbarium is a concept in which the digital images of herbarium specimens are prepared or stored in the databases. Yes, the digital images are scanned by resolution of 300 dpi and 6 dpi with high resolution. So we should have the high resolution scanner, and it is stored in the form of JPG or TIFF images, so that everybody can consult the digitized herbarium and can be used in identification and other purposes. And the virtual herbarium is just access of digital images in a virtual mode at any time. You can take help of this virtual herbarium anytime. You can carry this file. You can carry it anywhere. The access of digital photos is mainly helpful for remote users who can from anywhere in the world. Okay, the primary purpose of the digital herbarium is to reduce the damage of original herbarium. So you cannot open it, you cannot visit, you should not visit. If you not visit the herbarium, definitely you can carry the digital herbarium for using in the field. So as a genetic resource study, that yes, we have 20 minutes still. So as I just mentioned about the how to prepare, what is the use of herbaria, what is the specific feature of our this national herbarium cultivated plants. So what is the use? Now the herbaria is used in genetic resource study because we have information on the data on flowering and fruiting. See, it will help definitely when you have to visit. If it is the mentioned that fruiting time, definitely you can consult just in beginning mentioned that you have to visit the herbaria for knowing about the fruiting, maturity period, any other character or its identification. So it will help to plan the explosion of tour itinerary. It will help you in the preparing the tour itinerary, and also you can study the ecogeography versus morphological variation in particular ecogeographic conditions. What was the morphological variation there? So you can study from the herbarium. So it is an important source for study. Strategic and novel scientific activities in plant genetic resources, crop varieties, etc. You can make some novel scientific activities, and you can study the crop varieties linking distribution, locating areas, and mapping. Yes, it's a distribution. You can know about this distribution, area about its availability, and mapping. You can map it and you can visit that area from where it was collected for any germ plant collection, collecting species with narrow range of distribution. That is, these are the endemic cultigens, wild taxa, etc. That it will help you in collecting these endemic species, narrow range of distribution, wild species, what you are want to collect, and gap identification for germ plant, because it will also help you in identification of the gap, where gap is still there for collecting. If it is available now and good, that important material in our NSCP available, then you can make the visit after identifying the gap, visiting the herbaria. Bioprocess, definitely the ethnovertical observations will help you in this domestication study and many other studies. If you can see during ethnovertical studies, you can also record this medicinal use, extractive use, any other use, domestication. Sometimes what happens that tribal people, they collect material from important material, useful material from wild, and they are maintaining in their kitchen garden, because they have got some important uses. And many new this medicinal plants have been identified through the study of the ethnovertical in this information. So it will help you in bio perspective also. Then you can see here some they have the one paper is published by Dr. Kuldeep Tripathi and the other Dr. Ravi, and that on the Vigna vexillata, and they have recorded they have studied the herbarium, and also herbarium from this it mentioned Sikkim by it gives the ability of particular species since long time in Indian subcontinent. And again, another specimen from Khasi Hills of Meghalaya in 1951, both the specimens noted the edibility of tubers. So the information reported in the particular that herbarium will help you in the studying the important characters of that particular PGR, plant genetic resources, as is mentioned here. Then one of the important material collected by us from the this Halakandi district, where a specific type of this cucumber was being grown by local people, and has got the scaly epicarp, and it is an identification characters. So it is, you can see Mr. Ravi, he has just remove this part, which is the important characters, important key characters of that, and it can be traced on the herbarium sheets. So anybody can, if you want to see, you can record the ventral side and dorsal side also of that epicarp for the as a identification mark. This is this you can see the herbarium that photographs he has taken, and also he has removed, sliced the fruit, and also you can study the how the carpels in the fruits, its skin characters, and other things that you can mention. You can see here that how we can study through this the material collected from the this Hailakandi district. And then there are some problems we are just facing, that in case of the handling of this herbarium resources, that maintenance of RH of 50% and temperature of 15 to 18 degree centigrade is maintaining. It's very difficult. So these problems we are this facing. The sprouting of some species is you can see some orchid, if you are preserving the orchid, you can sprout in the herbarium itself with some having some moisture and other things. This if it has got the ideal condition for sprouting. Damage of seeds in the pods of legumes. Many times some legumes are the legume pests are very serious problem of the pulses. So it can be damaged by the these insects, and then sprinkling biovestine, naphthalene powder. It is also to avoid the pest damage. Again, very is not good that it can be, it can infect, it can affect your health. So regular inspection of specimen is again very important, very difficult, that needs to be done. So these are some of the important things we need to, any curator or any herbarium person should follow, or he can face, he should, it is bound to face by the curator these problems. So I just mentioned here, it is a important tool for the systematic studies that we have conducted, systematic taxonomic and systematic studies on many native crop taxa like Oryza, Vigna, Crotalaria, Cajanus, Microtyloma, Sesamum, Abelmoschus, Lufa, Trichogenthis, Allium, Moringa, and Osum. They had this native taxa studied. Taxonomic studies has been done, systematic study has been done by using our herbaria, and taxa of doubt are studied in detail for further validation using evidences from also some species, as mentioned, see, which are unable to identify through other evidences that biochemical molecular tools is used through the collaborative research of our other these divisions like molecular these genomic resources, then also our evaluation division, also from the field, our regional sessions, so that we have taken help for the clear the doubtful study of the material. And as mentioned, systemic studies we have undertaken on Herpetospermum, just mentioned, Northostordum gracile. It is an important weed, and Solanum diaphyllum again studied systematically, and also preserved these herbarium of those material. Systematic studies on indigenous alien species. Yes, it's a very good, very big. This material was difficult, that in India more than 30 species are available in the mostly in the Himalayan states. So the problem or identification was big in case of allium species, but our senior Dr. Anjula Pandey, she has done the taxonomic studies of these allium species and developed or brought a publication on the catalog of these species you can use. So what the method we are following for the systematic studies, we first select the crop taxa from the crop complex, taxonomic ambiguity, who having the taxonomic ambiguity, least work out groups. So these are the some of the area or the area or the species or the material where we just choose them and then start the taxonomic and systematic studies. And also for that purpose, we review the previous studies done, and then protologs, revision, monographs we consult for this review work. We start for this taxonomic studies by systematic studies, field studies, definitely for niche specific species in flowering, data on flower, fruits, and other identifiable traits is studied in the field. And herbarium studies, definitely it is required for initiating any this biosystematic study. And experimental studies is very important. You can grow it pot, you can experimental feed, you can grow it in experimental feed. You can also test in the labs for its biochemical things and other things. You can do it is required to do the experimental studies. And supportive studies also on molecular, biochemical, ecogeographical, cytological. Then expert consultation is again very important, because the expert consultation is very important to know about its identity, to know about its characters, to know about its the other things available in that particular Suppose biochemical study you are doing, so you know that what is the usefulness that is required for that purpose. You need to consult. And herbarium, that molecular studies also, that what is the DNA and other things which is helpful in the studying to any PGR. Then identification on morphological marker characters is important. Closeness between crop and related wild taxa is also studied. Identify the report the new species records, that is the one of the part. If it is a new species, you can report as a new species. And use in PGR management, authentication of gene bank accessions. If this systematic studies will help you in authentication of gene bank accessions. Sometimes some wrong names or particular names are mentioned in the this collection, so that you can help in the authentication of gene bank accessions, authentication of herbarium accessions, and identification. Also, the identification of crop quality tips. It will help you that also in phylogenetic relationship, prioritization of taxa for PGR management. So this is the flow chart on the activities of biosystem studies on taxa of PGR importance. So this flow chart is followed for any biosystem study. So this is one of the important material. It is known as ARIA, but earlier NBPGR no accession of this ARIA, that it is a Cocos Milo variety alvarensis. Now it was named as alvarensis because it was collected from the alvar. For that purpose, this material of all cucumbers were grown, and either in the net house or in the experimental garden, and help was taken from the evaluation division people, and they have helped, and then the systematic studies was done, and then a new species was described, that is the Cocos Milo variety alvarensis, because it was collected from that area. Earlier, we didn't have this accession not available in our national gene bank. So this is the one of the important collection made from the alvar, and it's very important for pickling, for as a vegetable, as a raw material, and other purposes also. It's a very important material that is collected from the alvar, and then compared with the other cucurbits. Then allium, as I mentioned, you can see many allium species collected from the western and eastern Himalayas, and these are grouped are in semi-cultivated, minor, less known wild types grown in the tribal areas of Himalaya, like the allium tuberosum, allium praegesculinum, allium hawkeri, allium strichii. This is one of the endangered species grown in the western Himalaya. So many this allium species were collected, studied, identified, and also we have developed a repository of this allium at our regional station of Bhavali. So anybody can visit the Bhavali center, where number of species of allium are available. Also, this material is available in our tissue culture repository. So in case of allium, there are so many species. It was studied through the biosystem study done in this crop group. So why NSCP? What is the difference between NSCP and other herbaria? It is a repository of crop taxa. That most of the this herbarium belongs to the crop taxa, wild economic species, crop wild types. Any other useful this material is preserved in our this repository, and these specimens are arranged as in family wise, then by genera, and then by species, and in alphabetical order we have arranged. This is different. Then otherwise, I just mentioned, is any in every herbaria, then we have to follow the this classification system, convenient for PGR workers, parabotanists, and non-taxonomists and beginners. It can help in this, and it emphasizes the crop plants and wild relatives. Okay, provides a unique HS number and provides authentication certificate, just as mentioned, and also develop guidelines for collecting, preserving, and then using this for the wide users. It is already available at our library, and also in our ebook it is available. You can visit our this web page of NBPGR and you can see the guidelines. These are the significant collections. Also this were collected, and the systematic studies and herbarium were prepared, like this Mogonia. In some less known domesticated species, Mogonia vestida is grown in this. It is known as Soflong, grown in Meghalaya in very limited pocket. Quaix lacrima jovi. It is a job steer grown. Okay, Malva verticillata. So these are the some of the important less known domesticated species, important types of crop wild relatives as mentioned, some specific collections of wild bigna, wild allium, wild TTC. TTC means from Western Himalaya, that wild relatives of wheat and barley were studied and collected and mentioned. These are the herbarium, some of difficult species, how to prepare herbarium for difficult species. As you see, Musa in abundances is preserved, how it was preserved. It is just you can see. And these are the significant. Okay. Hello.
Sir, please, sir.
I'll please.
Next speaker also joined. Please.
Okay.
Yes, sir. Yes.
Okay. So these are the some of the catalogs we have just prepared of type specimens. You can see, and how to deposit. This is already mentioned. You can apply for this your herbarium in the for deposition. It is mentioned here, and we'll issue the adjust number to the digital images of the specimen also mentioned, and guidelines you can follow. So we also train the local students, and in the Delhi also you can see we are just explaining about how to prepare the herbarium. And these are some of the significant documents, that wild relatives of crop plants, wild evil plants, wild relatives of crop plants, importance of watcher specimens, success stories. It is here. The new initiative we are now collecting the this material, that if any differences in the cultivated crops, we are collecting them from the experimental field, and teaching aids. We are developing field keys, identification handbooks, text to manual database we offer. With digitization of cultivated plants, we have just these are some of the new initiatives, and these are some of the stalwart that Dr. Harjan Singh, he initiated this type of study. And at NBPGR, Dr. Rosina Nair, retired now, Dr. Anjula Pandey recently, she has retired, and they are the actual curator herbarium. They have just maintained this herbarium of National Herbarium Cultivator. That's all. Thank you for listening.
And thank you, sir. Thank you very much, sir, for a nice presentation, and it is highly informative. And dear, I am requesting participants, if you have any doubts, please give in chat box. And now our next speaker is also online. And thank you, sir. Thank you, sir. Please on screen. And I am requesting Dr. Jyoti Kumari, coordinator of the program. Please kindly give the biography of Dr. H.D. Upadhyaya, sir. Please, ma'am. Please continue. Ma'am, it is not in presentation mode. Yes. Ma'am, please keep in presentation mode.
अनशेयर करके फिर शेयर करिए, अनशेयर
yes,
करके फिर शेयर
अनशेयर करिए.
स्लाइड शो करिए.
2 बार क्लिक कीजिए मैम, 2 बार.
Okay, ma'am. Yes.
Okay. Unmute करिए, unmute, please unmute.
Yes.
Yeah. Very good afternoon to everyone, all the participants. Today we have very eminent speaker, Dr. S.D. Upadhyay, who is the ex-director of the ICRISAT Gene Bank. So, sir was born in UP, and from the beginning itself, he was very much fascinated with the agriculture field. He grew up in the agriculture field, so he got selected his career in the agriculture field. He completed his PhD from the Pantnagar University, GBPUAT Pantnagar, and he did his PDF from the ICRISAT in 1980. From the beginning itself, so sir's first paper came in crop science in 1998 itself. After that, he has published more than 50 papers in the crop science. And in ICRISAT, he was as a ground beater since 1991. During that time, he released 27 cultivars, and it was released throughout the world in 18 countries, highly popularized. And since 1997, sir was acting as a gene bank manager there, and he was a director of the ICRISAT Gene Bank. During his entire period, he has developed several core collections in the mandated crops of ICRISAT, chickpea, groundnut, sorghum, pigeon pea, etc. And he is the pioneer for the concept of mini core collection. So he has developed mini core. First, he developed core collection, and for all the crops, he developed mini core, and in several crops. And those mini core is being utilized for the trait discovery, and it has been proved that it is containing very trait-specific germplasm, and it has been used for trait discovery as well as the molecular genetics study also. Then sir also developed 23 elite genetic stocks for different crops. At present, sir is adjunct professor at the University of Georgia, USA, and University of Western Australia. Also, he is looking after as an adjunct professor. And based on the sir's work, core collection, at NBPGR also we took up initiative to characterize our large number of accession in case of wheat, chickpea, and then recently we have taken up in like sesame, linseed, lentil, pea, cowpea, and other vigna crops. So based on that, we have also developed a core set in the wheat, and mini core was also developed. So we are very much thankful to the sir that he was the pioneer for our work also. So based on his work, we also got inspired and we started our work. Then sir has got several awards, like he's the fellow of American Society of Agronomy and Crop Science Society of America. So here some glimpses of picture is there receiving award. Then he also received Frank and Mayer Medal Award for plant genetic resources for his vast contribution. Then he has also received Crop Science Research Award and International Service in Crop Science. Then Harbhajan Singh Memorial Award of our society, he has also received because of his great work in the PGR management and utilization. And furthermore, he is also the elected honorary fellow of this society, prestigious society, and he is also a NAS fellow. Furthermore, sir is honored with a very great dedication chapter, that is Haridev Upadhyay Plant Breeder, Geneticist, as well as Genetic Resource Specialist in the prestigious Plant Breeding Review in long back 2018 itself. So we are very honored to have today him as a speaker in our lecture series. And he published more than 950 papers. So great contribution to the science, agricultural science, that was included in Nature as well as PNAS and several other journals. So sir, we welcome you for your lecture on the topic is core and mini core collection for efficient management and enhanced utilization of PGR. So certainly our participants or trainees will get benefit out of your lecture. So thank you, sir. Please, I'm sharing the screen. Please share and speak, sir, and enlighten the participant with your great idea, knowledge, and words, sir.
Okay. Oh, what happened?
सर नीचे शेयर स्क्रीन आ रहा होगा आपका।
सर ग्रीन कलर का बीच में है सर।
1 minute, अभी करता हूँ मैं जरा। Okay. Is that okay now? Share screen.
शेयर स्क्रीन पे क्लिक कीजिए सर और फिर फाइल पे डबल क्लिक कर दीजिएगा तो हमें दिख जाएगा।
I don't know what is happening. Oh,
सर शेयर स्क्रीन करने पे वो फाइल दिख रहा होगा, पीपीटी वाला फाइल।
I think I am pressing something inadvertently, something wrong. So share is, this should be here, share screen, yes.
Yes, sir. PPT, sir, already open. Is it open?
Yeah,
sir?
yeah.
ठीक
Share
है।
screen, is that right? Now, should I click it once again?
Sir, click the share screen, then file you might have seeing file, no sir, on desktop or somewhere.
It is there in my the same place. It is
हाँ,
there.
उसको डबल क्लिक करके सर।
Am I visible now to you?
Slide
You
is
are
not
visible,
visible,
sir.
sir.
Okay. So let me open this again. Is this visible now?
No, sir.
Ah. Why? Okay. So first I click this, isn't it? Share screen.
Share screen, yes, sir.
Okay.
Sir,
Sir,
before
slide should be already open, sir, before.
Okay.
giving.
Okay.
Slide
Fine.
should be already open, sir.
Okay. I will do it again. Slide is open. The slide is open now. My
Okay.
PPT is open.
ठीक है सर। अब सर शेयर स्क्रीन पे सर you can click and then
That's what exactly I am doing.
it will go to that window where slide is.
Okay.
वहाँ पे डबल क्लिक करने पे सर फिर वो आ जाएगा।
Should I double click this thing?
Yes. Slide, sir. You need to double click slide, sir, wherever
Okay.
slide file is there.
Okay. Oh, okay. I got it now. Yeah, this one. Okay. Is that okay now?
No, sir, it is not coming, sir.
Oh. Why?
Otherwise, sir, you can send slide. I'll send from you.
No, sir. That I generally don't do. It is open. What did I do? Something is wrong.
What can we do?
let me again. I was listening to everyone.
Yes, sir. Sir, you close the file and again you open, sir.
That's what exactly
yes, sir.
I'll do. I will restart this every damn thing.
Yes, sir.
मैम सर से अगर वो फाइल मंगवा लीजिए, यहाँ से हम प्रेजेंट कर देंगे, सर वहां से बोलते रहेंगे।
नहीं, वो उन्होंने बोला ना कि जनरली ही विल नॉट शेयर, तो शेयर नहीं, कोई
वो
नहीं।
तो वो तो मैंने बोल दिया और
मैम उनको एक बार फोन कर लीजिए, चाहे फोन से समझा दीजिए कि पहले वो पीपीटी ओपन कर लें, देन शेयर स्क्रीन करें।
आई थिंक लैपटॉप रीस्टार्ट किया उन्होंने,
हाँ,
दिखते नहीं।
अच्छा।
Yes, sir, it is visible, sir. Okay, sir. Yes. Sir, your voice is not audible.
Okay, sir, come on screen, sir.
Yes, sir. Now it is coming, sir. Yes.
Okay, sir. Now you can start, sir. Okay, sir. Yes, sir. Slides are visible, sir. You can, I think you are mute.
Sir, please unmute your speaker.
Sir, sir,
Yes,
please.
microphone. So,
Sir, please unmute, sir. Your voice is not coming, sir.
Right, start
Yes.
video.
yes, sir. Yes.
Is it
Yes,
okay
sir.
now?
Yes,
Ah,
sir.
yes, sir.
Okay, sir. Perfect,
Is it
sir.
okay now?
Yes, sir.
Okay.
Mhm.
My apologies for this mess up. And
No,
in fact, I was ready for quite some time, but then
Yes,
anyway,
sir. Yes, sir. This
thank you.
happens, sir. Technical.
Yeah, thank you, Dr. Jyoti Kumari, for rather elaborate introduction. And how many participants are there?
Sir, total 84 are there, sir. 84.
84.
participants. Yes, sir.
84. Okay, so good afternoon to every one of you. First, I was thinking if there are half a dozen or dozen, we can have 10 seconds each, but each one's name. But then I don't think it's worth really going for that. Anyway,
Yes.
Dr. Jyoti Kumari told I am Haridev Upadhyay, and my co-author is Professor Peterson from University of Georgia. So I would be talking a bit about core and mini core collections for germplasm utilization and management. So this is the topic. And how do I go for changing the slides? Page down?
Arrow, sir. Arrow down. Down arrow you can do.
Okay.
Keyboard.
I am having keyboard, but then.
Sir, just first click on the screen. After that, you just use your keyboard.
First click on screen here.
Yes, sir.
Yes, sir.
Okay.
Then down arrow, sir.
Oh, good. Thank you very much.
Okay, sir.
So we all know the kind of challenges to global agriculture, including that of Indian agriculture. These are very old figures, some of the things I would be talking, particularly in terms of germplasm numbers, because they keep on changing. But yes, there is a projection that there could be close to 10 billion people by 2015. And a lot of problems are going to be particularly in South Asia and Sub-Saharan Africa. And that's because of climate change and other factors. Another socio-economic factors in these two regions, South Asia and Sub-Saharan Africa, because the risk absorbing capacity of the farmers in these two regions is very, very low. And that's why the breeders have a challenge to develop cultivars which are climate resilient, and then the use of those cultivars with management of natural resources can perhaps be of some help. This slide I always present in almost all of my presentation, and reason is very simple, that this talks about how and why we should have germplasm diversity. What is the need of germplasm diversity? There was incident in 1970s and in US regarding southern corn blight, and it reduced corn production or maize production in corn belt of the USA. And this is beautifully described in a paper by Professor Tatum of Sprague and Tatum of combining ability, who gave the concept of combining ability. And then scientists realized that we need to have germplasm collected as much possible. And why they thought that they should as soon as collect the germplasm, because or most of you might not be born by that time, but in 66 or 68, I mean, prior to 1970, a green revolution was happening in these countries, particularly which are the rich genetic resources, rich in plant genetic resources. And thousands and thousands of the land races were being replaced by single uniform varieties in the districts, entire districts. I remember, I come from Aligarh, and in our farm itself, we got some 1 kilo seed from Pantnagar, wheat seed, S27, S222 or 227, something variety was there, then Larma Roho from USA and sorry, Punjab and other places. So our entire field was sown with those varieties. So villages after village, we are adopting high yielding cultivars, and it was necessary. Otherwise, we had a problem in feeding our population. So there were two incidents, the germplasm uniformity, which caused a lot of problem in USA. So scientists recognized there, and then they also recognized that this diversity is going away sooner than later. And that's why Rockefeller Foundation and other institutions started collecting germplasm collection. As a result, we have close to 7.5 million germplasm accessions conserved in about 1,800 gene bank. This is about 4 or 5 years old data, and new germplasm numbers and others will come in status of world plant genetic resources, which will come from FAO, I think in 2023. You can see one thing, that only 25 to 30% of them are unique. A lot of repetition is there, because the line we have in ICRISAT gene bank, NVPGR would have, several other gene banks will have. So everybody counts it. And there is no way to find out duplicates and other things among different gene banks and things like that. So only 25 to 30% are unique. And crop wild relatives are about 10% of them. Among crop groups, again, you can see cereals, about 45%, food legumes, about 15%, and vegetables 7%. 11% of these total germplasm collections are in CG gene bank, including that of ICRISAT. ICRISAT gene bank, where I have worked for close to 28-30 years, has about 130,000 accessions of its mandate crop in 6 small millets from 144 countries. 111,12,000 have been safely duplicated. So this is how we think of utilizing the gene bank diversity, which can be used to develop the improved cultivars to fight malnutrition, improve productivity, and meet the climate change problems. So the question comes there, what we have done so far with this germplasm collection, which we had at ICRISAT. We distributed close to 1.5 million seed samples, 107 germplasm lines directly released as cultivar. Just take the germplasm line, evaluate scientists in different countries, evaluated in 53 countries, and then more than 1,000, maybe at present about 1,200 lines have released as cultivars. How a single line can change? Those of you who are involved with the legumes or pigeon pea can see the contribution of a line, ICP 8863, which was collected in India. And then this line did a lot of wonderful things when it was released for cultivation in Karnataka in 1986 as Maruti. It's a wilt-resistant line, small seeded one. And then the estimates do indicate that in the 10 years itself, it has given value worth $75 million, which is about 73% of internal rate of return on the expenditure. And here is a web link where you can see a story done by Crop Trust on this line. Then this question is that are our germplasm collection complete? We continue to collect them. Of course, they are not complete, but is there any way the collection are targeted? I mean, of course, whenever we put any proposal, we say, oh, we are going to collect. You can go to farmers' field and collect as many as you want. But is it based on sound scientific analysis or just heck of it? Still, you go to anywhere, whether farmers' field or market yards and like that, you can collect the same sample from anywhere. We did some studies and on sorghum in say different regions, South Asia, East Africa, pearl millet also, chickpea, pigeon pea, and groundnut also. These were the one where we did indicate that, okay, these are places, these are crops in which collection can be done, and then targeted areas, not anywhere and everywhere. And this was used for collection mission by Global Crop Diversity Trust when I was there in ICRISAT Gene Bank. I don't know what's going to happen and I don't care anymore. Anyway, let me go. Then the second, or rather third issue comes that all of you, I think, are plant breeder. Are they plant breeder, Jyoti, or they are?
Sir, most of them are plant breeder.
Okay.
Many of them are, sir.
Yeah, if you ask any plant breeder, including me when I was a groundnut breeder, if they ask, are you using sufficient germplasm? Of course, I am using sufficient germplasm. I am using more than what I need. That's a common refrain. And there is nothing wrong with that. It's truth also. They are using a lot of germplasm. But then, are they using adequate? There is a lot of germplasm available, but people don't use, including me when I was a groundnut breeder. We estimated based on analyzing the pedigree of breeding lines developed in ICRISAT in 25 to 30 years in two crops, particularly chickpea and groundnut, and found that even in ICRISAT, where gene bank is in ground floor and first floor are all breeding programs, so there is no problem of supply of germplasm or non-availability and that kind of thing. So less than 1% of the conserved germplasm in these crops have been used, or rather 0.5% or even less than that. Then going through the different countries, the popular cultivars. So this is for the breeding lines development, breeder development. Then out of thousands of breeding line, only 1 or 2 variety becomes popular. So in wheat, potato, and soybean, if you see the founding germplasm lines are less than 10, 75% in potato, 50% in wheat and soybean. Research are also there that very, very less germplasm is used. It means that most crop cultivars stand on a very narrow genetic base. Now, I will consider example of chickpea, which is very, 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 very good for us. That from 1978 to 2004, that's about 26, 27 years, breeding program at ICRISAT, 3,500 breeding lines were developed at ICRISAT. We analyzed the pedigree of all of them and then saw how many parents. So roughly 91 germplasm lines were used. So if you simply divide 3,500 by 91, so number comes about 30 times each germplasm lines. But no, that's not the correct. Because two germplasm lines, L550 is a Kabuli type from Ludhiana, and K850 is a Desi type from Kanpur. They were used 850 times. Then their derived lines were used hundreds and thousands of the times. Similar situation comes in groundnut, not only in India, but I analyzed in China, pedigree of Chinese lines, also groundnut line, and in USA. So it's crop after crop. So germplasm, the second conclusion becomes that germplasm use by the breeder is limited. Okay. Then the question comes, why germplasm lines are not being used? I asked myself really why I wasn't using germplasm. And even when at least there were 10 years, 8 to 10 years, when I was overlapping my job. I was 50% or 40% groundnut breeder at ICRISAT, as well as in Malawi, as well as looking after the germplasm of ICRISAT. So I asked why I didn't use germplasm. By then, I mean, you can tell lie to anybody, your supervisor or like that, or in meetings and like that, but you can't lie to yourself. So I asked myself why the hell I wasn't using. And the answer was pretty simple. I didn't believe the data which was available in the gene bank, because the data, there was good data, but the fact is that it wasn't related to the traits I was looking for. It doesn't give confidence I want to have, or any breeder would like to have, before using parent in his or her breeding program. So lack of reliable data on traits of economic importance which show high genotype into environment interaction. If I want somebody to tell me that, look, I am working on drought tolerance in groundnut, can you give me 4 or 5 varieties which are good drought tolerant? Or aflatoxin resistance? They will say, oh yeah, this line is available, we found this for report, and you get, and when you yourself test them, you don't find. And then there is a fight between these two people, and all kind of good relations are really spoiled. So That is the reason. It's not fault of breeder because they don't use, because they don't have confidence. And again, a germplasm person who maintains, say, 20,000 germplasm lines of wheat, Jyoti must be maintaining 15 to 20. I don't think she has enough money or resources to give valuable data on each one of them. No, it's not possible, humanly not possible. So in 1984, Sir Otto Frankel suggested a core collection concept. And then he suggested that if we can reduce the size of germplasm from thousands to hundreds, perhaps it can be evaluated, and then breeders can identify the parents and start using it. And I also got really happy to note that, look, let us start developing. So in 1997, when I first tried to work on the development of core collections and others, we realized what kind of problem we are having. So core collection is a subset of germplasm, which is about 10% of the total germplasm accessions. And then it should be representative. The confusion have that people say that it should have maximum diversity. No, it should have representation of the collection. And with that limitation, if it has diversity, fair enough. Otherwise, for maximum diversity, you can have only 10 accession top, 10 middle, and 10 bottom for any character. You can have it. But then that's not representative. Okay. I will not go here. So how to develop core collection and mini core collection? There is a bulletin wherein we have given the method how to develop core and mini core collection. Why this is not coming? Okay, now you can see that entire collection is there. You can develop the core collection based on good quality data, and from that you can then go for developing mini core collection. So considering all those methods, we developed core collection. First one was chickpea. We used 16,991 accessions and developed the chickpea core collection. And then following more or less similar or little different approaches, and like sometime using different characters and that kind of stuff, we developed core collection in all of them. And then when we developed it in chickpea 1 in 2001, we thought that, look, this is a wonderful thing. And then we developed at the same time, perhaps next year or 2003 or 2003 groundnut core collection. Of course, it was published 2003, but it was almost similar 2001 and 2002. Then we thought that, is it helpful to the breeders? Okay, you have developed core collection, you have published a paper, but what use it can be? So a lot of publications we did. Then the question we asked was, it is essential that or rather I can rephrase it, can core collection be evaluated extensively, replicated multi-location, so that breeders can get what they want? So we did it in case of chickpea. Chickpea, we have a core collection of about 1,956 accessions. We evaluated it at a CRESET, and you will be surprised that it costed us close to $10,000 to do one location, unreplicated evaluation. Only operation money in 2000 about 22 years back. And then you get the data for one season, unreplicated, and one location. I mean, I'm sure no breeder would depend completely on that data unless he has visited the field, he has seen some lines 10 times, 20 times, or maybe 100 times during crop season. That look, yes, this I can have mental picture and my notes say that this is good. So then question was, no, it doesn't help. It doesn't help. So in the second paper, when I wrote after that, of the regional patterns of variation, I think that was in Euphytica in the same year itself. I said, no, it's not going to help. Then it is not going to help. Then why? Because size of the entire collection is large. And that's why you need a core collection of thousands. My core collection of sorghum is about 2,247. 2,094 in pearl millet. Why sorghum breeders should evaluate 2,247 at 4-5 location? His entire money will go there. No, it's not that important for him. So then we thought that, look, what is the remedy? So in that paper I wrote, I think it was in Euphytica, that perhaps core of core, which we called mini core, is perhaps remedy, so that size becomes much less, about a 1%. So if it is in hundreds, you can do it and can get things done. Okay, let me know. I think it's 1 o'clock already, so I will not take more than 25-30 minutes. Is that okay, Jyoti, or you want me to? I don't want to extend your lunch time.
Sir, we have enough time, sir. You can continue, sir.
Correct.
1:30 tak you can continue.
Yeah, 1:30 or maybe 2-3 minutes early I will finish.
No issue, sir. You can continue.
So we proposed two-stage strategy to develop the mini core collection. First stage, develop core collection. This is available in my publications. You can find it. So these were the mini core collections we developed, and now you can see the numbers. Chickpea 211, peanuts 184, pigeon pea 146, sorghum 242, pearl millet 238. So it's in hundreds only. Then the second question, third or fourth question we can ask sequentially is that, okay, you developed core collection, but does it help does developing mini core collection helps identifying parents for the use by the breeders? Because again, it gives me a lot of publications. My CV may be good, but is it useful to the community, global community of breeders? This was the bulletin I was talking. This is information bulletin number 78, where we have given this. Now, I will come to that whether you can identify parents which are needed by the breeder. But one question which I faced in 2000 to 10 years, 2000 to 2010 really, that look, Hari, you have developed core collection by using these traits, but does it have molecular diversity, enough molecular diversity? What would have happened have you used the molecular markers? So my answer was that, look, I don't know what might have happened, but the fact was that I have only these characters data available. Only we did it, we recollected some data which was missing, and we didn't have molecular data, because at that time, one SSR marker point was costing us $1.5. So if 20,000 or 17,000 chickpea was to be characterized with 30 markers, it would have costed us $1 million. And that kind of money nobody has at that point in time. So answer was that when we start working with the Generation Challenge program, we genotyped our collections. So first thing is we developed composite collections in which core collection of each of the crop was involved. So chickpea we genotyped, chickpea and sorghum about 3,000, groundnut, pigeon pea, pearl millet and finger millet 1,000, foxtail millet 500, and 20 to 50 SSR markers we used in genotyping that. We have core collection, we have mini core collection. So now these 3,000 chickpea will consist of 1,956 core collection and 211 mini core collections also. So then from these 3,000, we took a sample of 300. So suppose if I consider chickpea 3,000 as entire collection, and then from this I want to have 10% core collection, 300 accessions. So this is exactly we got it. Now you can see things over here in on left side. This was 2,950 15 chickpea composite collection, which has core collection also. So you can say that Desi and Kabuli are roughly different. There is one mega cluster which is mostly of Desi, another one of mostly of Kabuli. And I remember that when I was working on this paper, this was in I think BMC Plant Biology in 2008 or 2007 it came. So I was working in this paper and I was visiting my village in Aligarh. Oh, and one of my farm, my elder brother was looking after our farm, and we have some people also working. So my elder brother asked me, "What are you doing?" When I was working on my laptop, I said, "Look, I told him I'm working in Hindi. I told him that मैं chickpea पे काम कर रहा हूँ." तो then he asked, "What does it mean?" I told him, "This is Desi, this is Kabuli, and like that." He said, "Yes, this is Desi." So he asked me, "How much money you have spent on that?" I said, we got $150,000. So he said that you can see with eyes that Desis are different from Kabuli. So why the hell you should spend that much money? So his version really struck. But then that's a fact that, okay, you see that chickpea, Desis and Kabulis are different. Here was the evidence which helped us to say that, look, what we developed, core collection or mini core collection based on phenotypic trait, were not inferior. Say if composite collection, 2,915 accessions, based on 48 SSR markers, captured 1,683 alleles. And then a sample of that, 300, based on 48 S marker, captured 1,350, 78.1% allele. And when we use data only on 7 qualitative traits, which is very simple traits, flower color, leaf color, and that kind of stuff, you still get about 74% of alleles. And when you use both SSR and qualitative traits, you get about 81% alleles. This was same thing in I think groundnut. 95% allele, 93%, 96% alleles, 14 qualitative as well as SSR, 20 SSR markers. So this demonstrated that whether you use morphological trait, such as even qualitative traits, or even molecular markers, similar allelic richness you can capture. I didn't have answer in 2001 when I was invited for a lot of lectures in Crop Science Society of America meeting or different universities like Georgia, California, and others. I didn't have answer at that point in time because I didn't have data to show. But in 2008, when we published this paper and when I visited again, I was talking, and then I said, look, gentlemen, we can now say, and this created quite a furor in South Korea. I was giving a presentation in the conference, crop science conference, wherein there were 65 genomic people attending this, and all of them said, Hari, this cannot be true. I said, look, I can give you data of both traits. You do yourself, and then let us see if it cannot be true. So then we published this, and then it gave the answer. So now question was that this was the main question, that are mini core collection useful in meeting the needs of breeders and other researchers, not only breeders, but other researchers also, pathologists. Can they get the sources they are looking for? Is it useful to the genomic people, the mini core collections and others, for GBAS studies and others? So now first let's look what breeders are looking for. So I used this term at that point in time, trait-specific germplasm. So somebody in UK asked me, Hari, what do you mean by trait-specific? I said, look, trait-specific germplasm is the one which you are for a particular trait. It could be for one or two traits also. Then second point breeders are looking, they are not looking the resistance or other things in a very poor agronomic background, because if they use a poor agronomic background line as a parent in their breeding program, then their breeding program doesn't progress as good as it should. So trait-specific germplasm, then good agronomic background, or rather good yielding background, you can say, let me say. So we used it in all crops: chickpea, groundnut, pigeon pea, sorghum, pearl millet, finger millet, foxtail millet, proso millet. But in this presentation, I'll consider only groundnut as an example, because I took it through as a breeder also. If some of you don't know, this is the groundnut you have. So we started identifying sources for tolerance or resistance to different stresses. And drought is considered one of the problematic abiotic stress. And then this variability for drought resistance related traits in the mini core collection of peanut, I think in 2005, we identified about 18 accessions which were good for traits related with drought tolerance: transpiration efficiency 10, spade chlorophyll meter reading 18, water use efficiency 10, root length density, total dry matter, and then intermittent drought tolerant from mini core only 184 lines. We got 11 lines. Salinity tolerance 12, and low temperature tolerance 158. This I remember I presented in some of the meeting in NVPGR when Dr. Gautam was the director long back, I think 2002 or 2003. Late leaf spot resistance and rust together 6. Early and late leaf spot 4. Early, late leaf spot and rust 3 together. Bacterial vect doesn't come over here. It comes in East Asia, Eastern Asia. So 7, aflatoxin 7, pre-harvest infection. Then sclerotium blight 67, and some of them were done in USA. This Chamberlain and Maluk in 2011 in Epres, they presented this thing. Then protein content, 18 accessions. And here what we did was that along with trait specific, we evaluated these mini core for I think 7 seasons. And you can see that out of these 18 accessions, which were good for high protein, 3 produced 1 to 5% on an average more yield than the best variety Icrisat has developed at that point in time. So we used the control cultivar and we used the best cultivar for yield comparison. Similarly, oil, 18 lines, linoleic acid, oleic by linoleic acid ratio. And then there were 21 mini core lines, which were better. 7 of those lines, multiple. I will show you these 7 lines data. They produced 2 to 37% more yield. And not one season. 7 seasons, rainy as well as post-rainy, both seasons. Iron, high iron lines, high zinc lines, then both iron and zinc together. So now you have biotic stresses, you have abiotic stresses, you have iron content, you have zinc, you have protein, you have oil, you have oleic acid, you have linoleic acid, you have oleic by linoleic acid ratio, you have all traits on them. And then over and above, at that point in time, we also have the molecular marker data on 20 SSR markers. Okay, let me check time. Yes, it is going. So if you see that paper, this slide has been, this PPT is taken from the paper itself. So these are sources and these are references where these lines have been reported. Okay, so you can identify now that 28 lines out of 184 were abiotic stresses tolerant, and 3 of them produced more yield than the best control cultivar. 30 biotic stresses, 16 nutritional quality traits, both iron, zinc, oil, protein, everything put together. And then the lines which were nothing, neither good for nutritional, nor for abiotic stresses, nor for biotic stresses, for yield alone, if you consider, 18 were lines for agronomic traits, and 7 of them yielded good yield. And 9 multiple trait lines, which has both biotic, abiotic, agronomic, and nutritional trait. And 4 of these yielded 3 to 33% more than the best control cultivars. Okay, so you can get what you need, provided your subsets like core and mini core are representative based on good data. No need to hurry things. These are those lines. Now you can see, this is 9 seasons pooled data yield, 3 rainy and 6 post-rainy. And this was paper published in 2014 in Crop Science. And you can see some lines yield as high as 2.5 tons. Then what we did was we identified based on SSR markers, you can see the diversity, which lines are diverse. So national partners, do they get really benefit out of this? I get benefit because I go and get appreciation by talking with the people. Oh, sir, you have done wonderful work, something like that, or publications and things like that. But are they used by the national partners? So these are some of the countries where this germplasm has gone. Now, total over 470, 480 sets has gone to 38 countries. These are some of the countries. So the question was then, that, okay, you identified, is there any greater use of germplasm as a result of mini core? Again, then it has to be verified by the pedigrees. So we did some pedigree analysis in chickpea. And we found that 2005 to 2008 at ICRISAT itself, as well as in groundnut, there was greater use of germplasm in the breeding programs. So that, okay, this gives what breeders need, and then breeders use. It's not, breeders are not resistant to the idea of using the good germplasm lines. Now, this question again came to me, that what happens if I use the germplasm lines based on my study from the mini core collection. So oil line we used. High oil line, remember in one of the publication we published high oil lines. So what happens if we use high into high or normal is to high, that kind of jugglery we all breeders do. So we used, we made some crosses and like that, and from the lines, and then we went up to F7, F8, RILs, and then identified 80 lines. Groundnut doesn't have more than 46% oil, 46 to 50% oil. Then up to 63%, and let me tell you, we tested this oil content not by NMR and NIRS also, as well as Soxhlet estimation, because reporting something having 63, more than 60% compared to 46% needs confirmation and reconfirmation before you open your mouth. Otherwise, it can make you a ridiculous person. So we tested this. So there are about 80 lines which has more than that. So if you can identify parents from a subset which has been developed, evaluated, then evaluation data you are confident, then you are analyzing, then taking through F8, F10 generations, and then testing those lines, and then testing oil content, not 1 or 2 seasons, but 2, 3 seasons, not by one method, but 2 methods. Because when I got anything more than 55, I will jump. I said, this cannot be true. Let us send it again, sample by different name. Then from laboratory, the same thing is coming. Anyway, so this has been, for sorghum example, I will say that mini core collection as an association mapping panel. So these are some of the publications wherein we have used mini core collection as an association mapping panel for different traits. And our recent one is in 2022 Journal of Experimental Botany, where we identified for biomass, sugar yield. So this gives you a lot of things to do. But again, question is, it gives you leverage provided your initial material is okay. So my conclusions are, you can read them, that we all know that germplasm use is needed. When at Pantnagar I was studying my MSc, my teachers will always tell germplasm is basic to crop breeding and crop improvement programs, blah, 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 like that. But really, when somebody start using, and in the kind of constraint we work to use germplasm, okay, once you use, then most people start using the breeding lines developed from that germplasm for that trait. Say, for instance, I was involved in breeding early maturing variety. So we initially used Chico as a early maturing source, and after that we were using derivatives of Chico. Why the hell I should use Chico? And it was giving me good early maturing lines. But then when we evaluated core collection of groundnut, we found that there are 23 or 24 lines which are similar to Chico and Gangapuri, but better yielding, better agronomic. So if they use, then you can broaden the genetic base of cultivars. So low use of germplasm is an issue, and core or mini core collection play an important role, particularly mini core because its size is less. But if somebody is having only say 1,000 core, 1,000 accessions, then you don't need to go for mini core because your core would be 100 lines or 110 lines, 120 lines. Then no, mini core is a thing when you have thousands and thousands of the germplasm lines. And then both molecular markers as well as phenotypic trait data can very efficiently extract a set which is representative. Minicore has been recognized as an international public good, and I say Minicore works. And with that, I thank all of you, and I do acknowledge all the people in 20-30 years who has worked with me at ICRISAT and different universities in the US as well as in India. And my co-author, Professor Peterson, and all colleagues in NBPGR, whom I had very good arguments and sometimes not so pleasant arguments last 10 or 20 or 30 years back. But anyways, that's part of the game, a part of our profession. Thank you very much. And if there are any questions, I would be happy to try to answer. And please don't use things from here, because if there is a typo or something, better go to the original paper. See, I might have done some mistake by typing something somewhere. Okay, thank you very much.
So I request participant to ask a few questions. As sir has told, sir can take up a few questions. So if you can raise the hand, or you can put in chat box. From there also we can take up. So the lecture was very good, as most of you are plant breeder, many of you. So this concept of core and mini core collections. So we learn from here that what is the requirement of making core and mini core collection, how it can enhance the use of germplasm by the breeders, and what are the impact of mini core development on the variety development, and then impact overall economical aspect, and then sustainable development of the crops. So if anyone has a question, so you can ask here.
Hello, madam.
Yeah, please. Yeah.
Madam, jo many germplasm, so many duplicate. So how it possible to remove one by one so many similarity of germplasm also during the collection?
Yeah, sir will answer. Yeah.
Look, this is a question most breeders will ask most germplasm people. My priority, okay, we know that we have germplasm collection like GL24 or TMB2, 10 accessions of that, or 20 accessions of that. You can remove that by passport data. But then I tell you an example which will say that, look, this is not really needed. When ICRISAT had the chickpea germplasm, ICARDA was established. Okay, ICARDA took ICRISAT's germplasm of chickpea. So we have the sample of chickpea at ICARDA, same line. We have sample of chickpea at ICRISAT when we got it from somewhere, Rockefeller Foundation or somewhere. And then when the same sample was sent to ICARDA, after 20-30 years, we got back the same sample. We did this study in GCP program. We got the same sample back, and then it was genotyped, the SSR markers. Two samples were entirely different.
Sir, there is one question by Dr. Malar Viji Devrajan. I'm
Okay.
reading out. What is the strategy to develop core and mini core collections in uncharacterized large germplasm collections? For example, around 500 number.
Look, if you have uncharacterized germplasm, I do not suggest you at least characterize them one season for certain traits. And unless your data is of good quality, I can tell you one example that when in 1997-98 season, I started working on chickpea. And when we saw the 16,991 accessions we used, data on several accessions was not there. So you should have a complete and good quality data. Not only characterized, but well characterized. Then you can, if you have 500 only, why should you develop core collection? You can plant them those 500 in 1 or 2 seasons, and then can get really what you want out of that. You can then select 100 out of 500, not only 50. Uncharacterized.
5,000 he has
5,000. Oh, sorry, 5,000. My apology. Then you characterized at least. Without characterization, I wouldn't suggest. Because even if somebody suggests, I will say not no. I can argue with him privately. But while answering the question after my seminar, I will say it's not a good idea to develop core collection from an uncharacterized germplasm.
I want your opinion, sir. Whether characterizing all 5,000 at one location is good, or characterizing 5,000 in fragmented way, means 1,000 at 5 locations or 5 years, that is good. Actually, at NBPGR, we have taken up entire at one go. But at ICRISAT, in some study, I have seen multi-year characterization.
That I can discuss with you one-on-one. I cannot answer now because I have some very strong views. You know my views.
Yes, sir. Yes, sir.
On public platform, no, I will not.
Okay. So if there is no query now, so we would like to thank sir, Dr. SD Upadhyay sir, for a very nice lecture. So certainly.
Madam. Hello, madam.
पूछना है sir? Yes, sir. Tell.
मैं रमेश हूँ, मैं PJTSU.
Okay, sir. Yeah,
Hello.
please tell.
Hello.
Please tell, sir.
Sir, one doubt, sir.
Okay, sir. Please.
Madam, when characterization the germplasms, generally, preferably, which character is important? First, foremost, qualitative or quantitative? That's my doubt.
Okay,
from
sir.
the expertise, sir. For most important, required to depend on area to the different interest is there. In general, which one is important?
Sir will better tell. In my opinion, qualitative traits.
No. Look, these are, we have certain descriptors, and those descriptors for each crop, at least our mandate crop, has been developed in consultation with the crop experts. So those descriptors we are using, and both qualitative, quantitative, agronomic, every damn thing we are considering there, with whatsoever is available in descriptors. So it's not my view or somebody else's view. It is the collective view of global community of that crop, which tells that, look, these are the descriptors. It may be qualitative, it may be quantitative, it may be yield agronomic traits, it may be quality traits, different traits.
Yeah, even comparison has been made in papers also. So both the traits together is good, qualitative, quantitative, as well as passport data also you have to include.
You can use anything information. First is your passport data should be correct. Passport data available should not indicate that this line was collected from Pacific Ocean or Indian Ocean, middle of the Indian Ocean, because sometimes passport data is so horribly wrong. So checking data is important. Of course, the thing which was raised by the first gentleman, that okay, what about the duplicates? We should use only one version which is that line. Say TMB2, if I have 39 or 24 accessions, we were using a particular line as TMB2 in all our things.
Any more questions?
Thank you, sir. Valuable information.
Thank you,
Thank
sir.
you, sir.
So, I would like to thank Sir Dr. Hari Upadhyay, sir, for his well-organized, thought-provoking, knowledge-enhancing lecture about core and mini-core collection. So certainly all the participants might have gained the knowledge about the requirement of making core and mini-core collection, how it can enhance the use of germplasm in their breeding program, and what is their impact. So certainly all might have enjoyed the lecture by sir. So we can have a great clap for sir for this very nice lecture.
Thank you.
Thank you, sir.
Thank you, Jyoti.
Thank you very much, sir.
Thank you. Thank you. And have a great lunch.
Thank you very much, sir.
Bye.
So, bye, sir. So the next afternoon session from 2:30 will be taken up by Dr. Rabi Pamarthi for the next lecture. So have a nice lunch to all the participants and please come back at 2:30. Sir, thank you. Thank you one and all.
Thank you, madam.
crops, 21 major crops. He involved in diversity distribution and trait-specific germplasm mapping of indigenous collection. And we can say proudly he is a person who initiated the GIS activities in the Bureau, and he given that output is very good, and GIS pioneer, we can say, is in agriculture, horticulture crops. And he is a reviewer of the several journals, including Current Science, Canadian Journal of Remote Sensing, IJPGR and IJTK. And he is a recipient of CSR Pool Scientist and DST Young Scientist Fellowship. And in 2005, he got a SK Seth Prize by FRA Dehradun, and he already he visited abroad, Japan and Malaysia, and during several times. And he is having publication of more than 52 research papers and other bulletins and other popular articles and other proceedings or whether letters, anything. And he is a person, he is involved completely in my institute, is GIS activities, and the lecture includes the practical session. And this is very interesting and very useful in our work and in research papers also. Now I am giving the dice to Dr. D.P. Samval. Sir, please.
Thank you, Ravi. I am sharing my slides.
Yes, sir. Sir, full screen, slide mode.
Thank
not
you.
Visible, sir, not visible. Slides are coming, but it is not in PowerPoint mode. Full screen mode, sir.
कौन पहले स्लाइड ओपन कर लो, उसके बाद शेयर करो. First open slide in the desktop, then share screen. Okay, now it's fine.
Yes, now it is visible. Yes, yes. Please.
So this one.
And sir, once and along with the symbol, sir, Dr. P.K. Malav is also associated with this work and during practical session. Sir, please continue, sir.
Thank you, Ravi, for giving the nice introduction. So I will be talking about the GIS application in PGR management, and my good afternoon to all dear participants. Nearly 53 participants joined this talk. So briefly, I will discuss about what is the PGR, why we are using GIS technology in PGR management. Then I will discuss about the other technology associated with GIS. So first, I will brief about why we are using GIS in PGR management. And then I will discuss little bit all technology related to GIS, the system. And then we can start the practical part online, how we can use the GIS for mapping, diversity mapping, georeferencing, geotagging, all these things. So hope you will be find this lecture very fruitful. So see, here I will not discuss because the morning session, our two senior colleagues, they delivered the lecture related to exploration activity. So I want to just discuss about what is the PGR, because PGR is the main focus of the lecture. So PGR are most valuable and essential basic raw materials to meet the current and future needs of crop program of the country. So our country needs so many PGR, means many crops, variety, all these things. But we are focusing for the PGR crops, which has the immense value. The PGR as a components of agrobiodiversity cover not only the whole gamut of genetic resources, but from advanced cultivars to primitive land races. We discuss about the primitive land races. From these land races, primitive land races came from the wild habitat and then from the primitive land races, then land races, then so many hybrids, all these things, domesticates, semi-domesticates, wild and weedy relative. This is the difference between PGR and other traditional university. We are covering all these things in PGR management. The diversity of ecosystem and agrosystem within landscape that are exploited in some way for agriculture and forestry purpose. This is our basic requirement. And just for this introduction, I want to highlight here, Indian Gene Center is one of the 12 mega biodiversity hotspot of the world. And out of the 34 hotspot of the world, India has 4. I can assume that everyone knows the Himalayas, Western Ghat, Indo-Burma, and Sundarland. Sundarland we can call the part of the Nicobar, Andaman and Nicobar Island. And this, for the PGR point of view, 730 species of crop varieties are listed in from Indian region, which is published by our division scientist in 2014. So why this is the topic of the today, why GS technology is very much useful in PGR management. So what we are doing here in NPPGR, our main focus, we have the data last 4 decades, we have the database, PGR database, collection, conservation, all we have the database. But what is the importance of that database in gap analysis? So here we are using the gap analysis technique for particularly georeferencing of the collected germ plots. We have the more than 6 lakhs accession in our gene bank. So we have the focus to georeference all the collected germ plot to know the diversity collected and the status of collection and conservation of the data. Mapping of germ plots collected versus conserved. So I will show those 2 maps where we have collected particular species and also conserved. And diversity distribution and species richness mapping, that is very important. Suppose we have the more than 1 species, we call the diversity in botany language. So we have so many species in sesame, wild species, solanum, and wild rice. We have the 7 species in India. So in brassica species also we have mapped. So I will show those things also. Mapping of trait specific germ plots collection sites and location. We should know from where we have collected, we have identified potential germ plots, trait specific germ plots. That we should know from which part of the country we have collected such precious germ plots. So this is very important objectives of our institute, and variability mapping in collected germ plots. So we want to know, because for example, we have collected more than 110,000 accession of the rice. Rice, the botanical name is the only Oryza sativa. But why we have collected those 110,000 more than that accession from the whole country? Because the variability and how we are using that, because center of origin of rice is the India. So we have the variability in seed shape, color, or products, uses, and different uses also. That's why we want to take those variability also within the district, within that state, and country-wise also, region-wise. We are mapping the variability of particular crop, you can say, particular crop or species. So next we have started this work of habitat prediction mapping using eco-crop and bioclimate modeling. That is the new area we have started. And here we are using, for this purpose, we are using DevaGS. This is the open source GS software. Anybody can, any participant can download if they have the passion or interest to learn the GS. They can download open source. This is the open source GS software. And ArcGIS, we have the ArcGIS also for introductory purpose for the student or starting to learn how the commercial software, how the commercial software is people are using. That is ArcGIS. In INVI also we have the remote sensing satellite image processing software INVI, and Quantum GS also we are working on this Quantum GS also. So see, for if we are working for the GIS technology, so we should know about the first without, for example, without data layers, we cannot start the work on this GIS environment. So GIS is the very big environment. GIS software is the very big environment, and I will discuss each and every components of the GIS. But data layers is very important, because suppose we want any layers, any shape layers, or particularly shape layers of the country, state, districts. So this is very important. Without data shape layers, we cannot prepare. We cannot prepare the GIS maps, different maps. And remote sensing data also in digital images. If digital images are there, we can digitize. We can screen, we can overlay on those satellite data onto the map. And GPS also very important, because suppose we want to link, we want to link our collection site with our samples, germplasm samples. So coordination link between the samples and the location, that is the geo coordinates. We call this latitude, longitude of that particular area, latitude, longitude, altitude, and other associated this feature should be recorded for linking of the passport data or germplasm data. PGR crop data in tabular form like that. Passport data we have, morphological, descriptor, evolution, so many data we have. So these data are very important for analyzing the gap analysis, diversity mapping, all these. Soil types data, shape layers of soil, from published map we can take, and different sources we can use the soil types map. From published literature also. Now in software you can prepare your layers, different layers for analysis process. Digital elevation data also. Suppose you want to see any flat area in elevated mode. So you should have the contour intervals, altitude, all these things, basic data you should have. Then you can prepare your digital terrain model or digital elevation model. See, so for the dear participants, if you are new to this GIS technology, it is our responsibility to tell about what is the GIS technology. So GIS integrate computer hardware, software, data for capturing, managing all forms of geographically referenced information. Any information related to our samples, we should document those. And if you want to go through the NASA definition, GIS is an integrated system of computer hardware, software, trained personnel, linking topographic, demographic, utility, facility, image, and other resource data that should be geographically referenced in the sense of latitude, longitude should be there for linking of the those site data with samples data. So GIS allow us to view. We can view our data, understand data. We can ask the question to data how we can interpret this, and how we can visualize the data in many ways that reveal the relationship. You can develop patterns, trends in the form of maps, reports, and charts. But it is the, I can tell here, in open source GIS software, any open source GIS software, you will not get all the answers. But if you will purchase commercial software, you can have all the facility of to give the answer of all the questions related to GIS and geospatial technology. GIS helps the user question, answer, solve problem by looking at your data in a way that is quickly understood and easily shared to anybody. PGR management through mapping of collected diversity, prediction on diversity rich area, locating suitable sites. Different climate crop models, GIS technology can be integrated into enterprise information system framework. Yeah, a complete information system. We can look for that purpose. The GIS is the very big tools. So we should know about the components of GIS. How many important? Because in GIS, so many components, in small, so many components. But 5 important components are very important. like computer hardware, software, data, methods, and expert, you can say, or organizational setup, or you can say expert in GIS, or some knowledge, not like that. Anybody little bit knowledge about the GIS, he can start the working on GIS. So computer hardware, everyone know. Software, everyone know. Anybody can purchase from the open source and market. But their data is the most important component of the GIS, because if data is accurate or right, then your output will be always okay or right. Data is stored as geographic data, spatial data, and related tabular data, non-spatial data. GIS integrates such spatial data and non-spatial data by using database management system. This is very important, and everyone should go through the data curation. See, here in this photographs also, you can see how the GIS 5 components are important. And here you can just glimpse, this is the glimpse of the in pictorial form. So see, understanding GIS tools, overlay analysis. So we have the so many layers in GIS. For example, I will show in GIS last part of this lecture the how we can start importing GIS India shape layer in software, DevaJS software. So we should understand about the GIS overlay analysis, why we are doing this. And in this PPT, you can understand overlay is the process of taking two or more different thematic maps of the same area and placing them on top of another to form a new map. So you will get some information. If you have the time one data or time two data, and you can overlay those two maps, time one map and time two map. So you can get some new information. So overlay analysis is the process of combining data or information from multiple data sets to arrive at new information. And overlay analysis again completes with any data type, raster, vector, digital, but type of output results depend on the type of input. It is in GIS system or GIS technology, it is 100% is correct. If you are inputting any data, the wrong or right data, you will get the output on that the same way. So here in the example, forest patches, you have the one layer, and slope you have the another layer. You can overlay those layer, and that you can analyze that result being so your average slope for each forest patch. Like that you can imagine. In this second example, you can see different data. If anybody is working on this rivers or water bodies, water ecology, so you can prepare one map, and other settlement area, any other person can make this building, all these things. If anybody working in this botany or forestry or biodiversity, you can prepare vegetation map. And then you have, if 3 layers or 3 maps in digitally, you can overlay. But if you have the hard copy of these 3 maps, you cannot overlay this. That's why the DS technology is very pertinent here. We can overlay in this using the different software. We can overlay and integrate a map. We can see for decision making or planning or working for research, all these things. So data is the data in GIS. Data is the fundamental assets of any GIS system to function properly. There are 2 primary types of data that are used in GIS. Data sets are grouped into 2 different types, vector and array. And why we are discussing, because we have the germplasm data, lakhs of germplasm data of different crops. We want to link those data in a particular format. So in GIS system, coordinates, latitude, longitude in decimal degrees are needed for georeferencing. Earlier, we have seen in database, people kept degree, minute, second in this one, and in the different database files. And it is very difficult to import the all data in GIS environment. That we are unable to import those data in GIS environment. But if we will convert those latitude, longitude data, latitude, longitude data into decimal degree, so then we can use that particular data. Here, conversion of latitude, longitude, decimal degree can be calculated with the following formula. And we have the in system. If you will work on DevaGS system, DevaGS itself have the one geo calculator, and you can calculate. You just input this degree, minute, second, and you will get the result. You will get in decimal degree, like 30.40 or 78.9, and you will get. But here in physical form, if you have not the software, or you want to convert degree, minute, second into decimal degree, so you should know about the formula. The decimal coordinates is equal to this H for the hour, and D for degree, M for minute, S for second. And this is the formula. Anybody, you can take the help of the other also, how we can convert this thing. And this is when you will input all these in your text, the Excel file or data file, all in decimal degree, then you can import your file into GIS environment. This is very important. So here you can see we have the one map. You can see here, one digital value is there. Another is a 2 is the digital value. Another is a 3. Here in general, you can say this is the digital map of some topography or some features that we can call this digital map or raster data. We can call, and the raster data, if your computer screen is there, every computer screen or raster data is divided into grids, row and column. And if one row and column is there, one small grid is there, that we call in remote sensing, the remote sensing application area, that is the pixel. That one discal number for one pixel. That pixel is very important. Or if you know the area of one pixel or resolution of one pixel, you can calculate the total area of that particular area. So we can convert. You people, suppose you are in physically here in our system, when you will get the opportunity to come here and NPP chair, and you want to work on the physically with our so many computers or so many technology, you can convert this through this software. You can convert this raster data into this vector data. So here 1 means, for the example, this 1 means cropland and pasture. A little bit this less digital value, this is equivalent to 1, and in 2 is equivalent to lake, and 3 for the residential. So in GIS environment, this is the facility, this is the software application. You can convert this digital value into this one, and you can estimate the area. That is important for policy maker or any area statics. If you want to know the impact of that particular area, so you should have the area statics. So based on the pixel size or resolution of that pixel, what is the total area of the city? So for that purpose, we should understand, because GIS is very big environment. GIS software has the full of big environment. We can use remote sensing satellite data, digital satellite data. If you have the imagery or hard copy of the satellite data, you can digitize in your digitizing table. You can digitize those, and you can import those satellite data into a GIS environment. If you have digital image, then no problem. You can just overlay any district, any state, and India map over those satellite data, and you can work on further that. So you should understand about what is the remote sensing, what is the GIS, what is the GPS, what is the ICT. All these are very important, and together we can call the geoinformatics technology. So in simple language, the term remote sensing means means sensing, or sensing means observing from your distance without being in physical contact. with the earth object or feature, because satellite is moving above the ground 500 to 1000 kilometer orbit of the satellite, all the satellite of the world, 500 to 1000 kilometer, different altitude. They are moving from east to west. They are moving. So we should know this, the satellite sensor is so powerful that can detect the digital, all the energy which is captured by the sensor, that we can convert into digital form, and then we can convert into analog form or map form. As per scientific definition of remote sensing, the science of identification, interpretation, and classification of earth feature. Any earth feature means grassland, biodiversity, forest, river, any dam, any area, settlement, any habitation, city, anything. So radiation as a medium of interaction by non-contact methods using satellite sensor. So here we can give example for West if our participants are new to this technology. So I can explain here, your eyes also working as a sensor, because our eye is looking on different earth feature or anything on our table, on our garden, anything. But our eyes not directly contact with any feature. So sensor is like that. So that's why we are saying non-contact methods or without being in physical contact. So you can understand, you can relate our satellite sensor, we call the MSS, multi-spectral scanner sensor, that we call MSS, different sensor. So RS, remote sensing, we can divide into active remote sensing on passive remote sensing, because in active remote sensing, satellite can use this signal from emitted from aircraft or satellite itself. And passive remote sensing, sunlight is the main source of remote sensing. So this is the difference actually. So most of the satellites are this passive remote sensing, because they are rotating, they are moving east to west from 500 to 1000 kilometer above the ground, and they are taking energy from sun source, and that energy 10 to 15% energy trapped by the sensor, and they are converting all reflected energy into digital form, and then again this form. So you can have this you will understand, you can understand this is the way, but this is not the actual distance of this, is just this is a demo. And here you can see in passive remote sensing, here you can see the sun rays. Here is the example for one ray is coming from sun and interacting with the clouds, all the atmosphere, and some part is reaching to ground, and whatever the reflectance, so many scattering is there, so many things are there, and if 5 to 10% only reflected energy tapped by the satellite sensor. So, and again, you people can ask this question, the satellite sensor is receiving all the data from the ground for particular day, when satellite is moving above the our area, or for the example above the Delhi or above the Bangalore, they are taking data. But it is very interesting, in our country only one data reception center is in Hyderabad, and we have got the opportunity in IRA. Some part of data they have allowed to us, and IRA also getting some data for data receiving center for agriculture purpose. And in another active remote sensing, you can see, the satellite itself is like a radar satellite. A radar satellite itself sending some signals to the plant. Here you can see, and here is also the same principles of physics of remote sensing is there. Some scattering, some problem is there, and whatever the reflected part is there, see that again trapped by the sensor and again received by the data center. So this is the difference actually. Here you can see we are also in the verge of the purchasing this agri drone, and agri drone also is because satellite is 500 to 1000 kilometer above the ground. So suppose this drone technology came for different applications. For here, I will discuss only for our India is the more than 60% farmers in our country. So our farmers should get benefit of the technology. So see this agri drone. Now farmers are using this agri drone in our country for spraying nano urea, pesticide, and insecticide, and sometimes they can use for the spraying the water, all these things. See the tank capacity. Now you can see 10 to 15 or 20 liter capacity of the tank, and this is the picture you say. What is why people came from this satellite technology, then aeroplane. See second picture, aeroplane. Now the only 2 meter or 3 meter above the ground. Why this? Because drone is a miniature remotely pilot aircraft. This is remotely pilot because one pilot can handle this drone. It is designed to fulfill requirements for a low cost platform with long endurance, moderate payload capacity, and capability to operate without a runway or a small runway. So that is support in our farm, IRA farm, we are using this drone technology. So we don't want this runway or small runway because we have the in between farm, we have the already some small runway. We can use this one. Drone includes equipment of photography, infrared detection, radar, observation, TV surveillance. All these software you can purchase and you can include. It uses satellite communication link and onboard computer controls the payload and store data from different sensor instruments. So many applications are coming in UAV, and here this is just this the picture of the platform of satellite technology. Any software, or you can see any medium from the above the ground, like camera also. You can give the example of the camera. If you put your camera in tripod, that is the part of the remote sensing. So we have started our work, and our work we have started from aerial photographs, and from aerial photographs which was taken by the aeroplane, then remote sensing, then GPS technology, then we have so many geostationary satellite. We call that those we call geostationary satellite communication satellite above the 35,000 kilometer above those satellite, and this GPS satellite also 18,000 kilometer above the ground, and these other application satellite now like the ground based remote sensing also you can understand, because 1 meter plus minus 1 meter you can use the spectroradiometer. For spectroradiometer handheld, you can take that handheld spectroradiometer with computer, and you can take that with the help of the assistant, and you can take the readings of digital reflectance value from the ground also. That is also ground. We call the ground remote sensing, ground based. And here also you can see the one drone is moving the air, and different things, how the development came. And here is the operating altitude we have given, and ground coverage also. If why we can call why we have the GPS satellite of the 18,000 kilometer, because we want to cover more area. Like India also launched first time India launched GPS satellite in 2013, and now in India also has the 8 GPS satellite in the orbit, and we want to cover actually not India but Sri Lanka, Bhutan, Nepal, Maldives, Bangladesh. We want to help other neighbor country also. That's why if we have the GPS satellite, the altitude is high. If we have the geostationary satellite, altitude again high, 35,000 kilometer hour. And if we are working for the remote sensing technology, hyperspectral remote sensing technology, we have the, you can say 500 to 1,000 kilometer. Most of the satellite of the any country of the world, they are rotating, you can say moving, or from orbit, in their orbit, they are rotating from east to west, all these things. So in detail, you can see what I already discussed about the cloud, scattering part, all these things. This is just for understanding how the satellite is taking the digital value, or this images, or this picture, or all these things taking from the how the sun ray energy coming, all these things. This is the best example. And how the sensor is using. This is the part of this the EMS part, electromagnetic spectrum. So you see here in general, from the visual, this left side, you can in my with this PPT left side, all these UV, X-ray, all this wavelength range is the part, is the application of medical side. But we can focus from this visible to our microwave. And again, you can see radio and television, they use high frequency. So we are focusing basically this visible part to most of the satellite visible part to thermal infrared, and many radar satellite for microwave remote sensing, penetrating rocks or ground, all these things. Those are the different applications satellite. So radar remote sensing. So that's why I'm showing this EMS spectrum. So causes of spectrum with wavelength, variation in reflectance, biochemical, structural properties, chlorophyll concentration in vegetation. So how the different sensor or working for different this wavelength range. That is very important. It is depends upon the chlorophyll concentration of vegetation, soil, mineral, water, background, organic matter, all these things. I will show this in this picture how the different bands wavelength range they are behaving differently. So here I will show. Here you can see this is basically for the standard reflectance curve of the vegetation, and vegetation reflected mostly 45 to 50% in infrared region. So you can see the from blue, green, red, and infrared region. So here strictly mentioned here blue in blue color, green and red. This is important area. And near infrared, because in near infrared has the no color, but the reflectance is high for 45 to 50% of that total reflectance value, highest in near infrared. Any part of the world, if you are taking any reflectance value or satellite image, so this most of the reflectance in near infrared region. So I will discuss this why we are making this use in FCC, or I will discuss in later on. And you can see leaf pigment is in very pertinent in blue, green, red region. Then near infrared cell structure. So water content also, why the three dips are there, water absorption mode. See, and dominant factor controlling leaf and primary absorption bands. So wavelength range we call in remote sensing language the band 1, band 2, band 3, band 4, band 5, like that. So when we launched first satellite in 1988, India launched first application satellite in 1988. At that time we have only 4 bands, and we were depend on the Landsat data of USA, and we face so lot of problem during that time, because application satellite launched by USA in 1972, Landsat 1 they launched in 1972, Landsat 2 in 1977, 1983, they launched so many satellites. But we launched in 1st March 1988, we launched first application satellite. I'm not the physics background, or we are the application scientist. So we are application side. We are using satellite data to our application or agriculture, botany, forestry, biodiversity, diversity mapping, all these things. So this is just this one. So we can see this small this one. I already discussed these things. Spectral universe, just I want to discuss, because field remote sensing, you people can now the cost is so much reduced, because earlier we purchased for IRA and Indian Institute remote sensing, we purchased 70 lakhs, 80 lakhs spectral radiometer. Now the cost so drastically reduced. So spectral that we call the field remote sensing automatically save data as a compatible medium, display reflectance transparent data in percentage wise observance versus difference, means relationship between observance and wavelength versus all these things. Single and multiple spectral plots are available. Compare current scan against library, including the USJ. Actually, different crop library, the United States Geological Survey, they developed different library, means digital library, the digital value of that particular earth feature they developed, and we can compare for the like we are using in our crop characterization, we are using checks like that. No? So we can use this USGS library and access to more than 19 vegetation indices. I will show one indices, two indices here for 19 vegetation indices, because most of the people working on this biodiversity, botany, or natural natural resource management, all these things. So here I have taken this slide, this map from National Remote Sensing Center 2020 developed. So we can use in different purpose. Here is this example of spectroradiometer, and through this you can take the reflectance value. It will record in your computer database all these things. And laser is there. It's the point tip. One small laser is there. That is the instrument. That part I will not discuss because we are the application scientists. So see. So just little bit about the sensor. Sensor is like our eyes. I already discussed sensor. Here is a linear imaging self scanner. This one means what is the abbreviation? Linear imaging self scanner. This one we launched in 1988. And how the drastic change. That time we have the only 72.5 meter resolution. And then we have the 36 meter. And these are the only four I already discussed. Spectral bands wavelength range only four. B1, B2, B3, B4. So range is there. So you can have the range. See. So how the development came last, you can say 30 years. And we have launched high cartosat, high resolution satellite. You can say resolution the 0.8 meter means 80 centimeter. In panometric 280 in a 3.3, 30 centimeter, you can imagine. And in one time, we have the first country to launch the 4 meter resolution data during 1998 or 2000. And then they launched the 1 meter satellite data. So this is the how the India is moving for this space technology, and we are focusing for the application of satellite technology in agriculture, particular, and this PGR technology in particular. So see vegetation extension. Just in that glimpse, you can see the bare soil color is different, vegetation cover is different, and sensing stage is different. Why the vegetation cover is red? So I will relate this to the infrared, because we want to know vegetation reflectance. So that's why we can make the combination. Infrared has the no color, so we can input a red into infrared and blue, different combination we can make. And the software, it is the facility you can make in infrared channel. You can input that red channel, in red channel blue and green. That's why, and you will get the FCC combination. And always any part of the world, any FCC, if red color, light red color, moderate red color is there, that is for vegetation cover. Means maybe crop, different crops, forest, grassland, any vegetation. So this is mostly means more than 80% scientists of the world, they make the FCC to identify quickly vegetation cover during landslide, this one hazards, all these things, floods. We are making FCC. So this is this FCC of ST block. This is near to Nagpur. We during my training in Nagpur, I went there for 10 days training, and there is also very good facility of remote sensing lab, GS lab, and we prepared our group. We 3-4 participants. We developed this FCC, and now the Sentinel-2 data you can freely update. But resolution is 30 meter. That I can understand that the resolution 30 meter. But for understanding, you can, or analyzing any earth feature, you can use the Sentinel data, Sentinel-2 data, free of cost, and you can prepare. And without any much knowledge about the remote sensing, you can make the 3 classes here, or 3 or 4 classes. One is the red, that is the vegetation, because this is the FCC. One is the light bluish color, you can see. One is the dark is deep water body is there, and light blue, some water channel, all these things. And another, you can see another this color. So you can make different classes. So based on this, and I already discussed in different spectrophotometer can use 19 vegetation indexes. Here I will discuss only this one, this RVI, another the NDVI, very important for this vegetation mapping purpose. And different bands of a multi-spectral image may be combined to highlight, calculate the vegetated areas. One such combination is the ratio. Actually, this is the ratio of the NIR and red band. NIR we call NIR and R. This ratio is known as the ratio vegetation index near infrared and red band. So I can show this. See, this is the example. And most of the people, for accuracy purpose, people are developing NDVI, normalized difference vegetation index, and see formula. So you can compute any NDVI. And one example I can give, our National Forecasting Center, Mahabalonus in IRA campus, they are after each 15 days, they are preparing this NDVI map for crop acreage, crop condition in India. They are using, but they have so many facility to use the NDVI map for different purposes. So here you can see how we can make the application of those spectral reflectance. So here you can see different, you can also publish very good things here for, you can identify corn, cotton, soil, many things you can identify different reflectance value. Because here also in another, that is impertinent example to, because in India in 1980s, first experiment was on this disease, how the any plant infected with some disease, and how because I cannot distinguish the inside disease impact in the plants. So satellite sensor can identify those impacts, if this plant is disease plants or not. So it is, we started from taking data from USA. See here, healthy plants in general, you don't think about the formula, you just see 72% reflectance, this NDVI value of this healthy plant, and only 14%. This is in senescence stage, and when it will complete this leaf fall condition or mature condition 100%, it will come down again. So see, so how we can differentiate the vegetation. This is very pertinent example. And for the not the only our farm, our area or field, but global NDVI. One agency is there, they are using modest satellite data, and they are developing global NDVI. So here you can see, you have the very good field. India is also good map of NDVI. So you will feel only part of this Rajasthan or dry area. So you can compare. So that well-known area or desert area. So you in desert, not only hot desert, but cold desert also you can identify the Trans-Himalaya, Ladakh, all these regions, also the yellow color. So you can identify not only the small. Now here I want to give this advanced technology, hyperspectral remote sensing. So what is the difference in remote sensing and hyperspectral? We can distinguish more within the species, or late, wheat, advanced, like here wheat late vegetative, wheat critical, corn early vegetative. We can distinguish, because our aim is to identify, to distinguish from one crop to another crop. That's why scientists develop this hyperspectral remote sensing to get the more information about the vegetation crop, because still many crops we are facing the problem. Anybody other than agriculture, botany science, they are saying how you are identifying the crop in the field, because all crops are green, most of the crops pattern is the same. No, now we have the answer of that question. We have the hyperspectral remote sensing. See, barley late, barley early vegetative can identify. So some limitations is there, but science is going on, technology is developing. So this is just answer of that. And we can, one another, I already discussed, we have the 98 only 4 bands, then we make the 7 bands, 10 bands. Here we can use the more than 400 bands we can use. And but after those spectral range, we should identify which spectral range is very pertinent for the biophysical quantity mapping or biomass mapping or agriculture mapping. So this is just see. Now I will discuss about the GPS, because in georeferencing or diversity mapping, we should know about the GPS. What is the importance of the global position system? Is a satellite-based navigation system consisting of network of 24 orbiting satellites, and that are 12,000 nautical miles, or we can say 18,000 kilometers above the ground. All GPS satellites are there. And first GPS satellite was launched in February 1978 by USA, and they have opened this GPS for the civilian purpose in 1983. But many countries, and we received this satellite during 90s. This GPS which is launched by the developed by the USA, we received 90s. And first time I saw that GPS, a big GPS, in 1996 in Dehradun. So you can imagine. But the important is now the India also launched the GPS satellite 13, that we call Indian Regional Navigation Satellite System. First 1A, 1, 2 like that, and 8 satellites in orbit. And some people have the GLONASS also, Russian satellite. And satellite takes 12 hours to orbit the Earth once. It is taking 12 hours to orbit complete Earth in 12 hours. So these are some important, how we can use this GPS system for latitude, longitude, slope, altitude, and all these points, how we can use, and what are the use of the GPS system in PGR management. I will discuss those. So see, latitude, longitude of your particular position, one should record data in decimal degree and altitude value in meter. So because we have the facility in GPS, and we have in our division more than 10 GPS, we have our regional center, and we have the more than 10 GPS. And now everybody, mobile system, advanced the system, you can use the GPS system also. GPS show 4 satellite points. Suppose you have the physical GPS, small mobile. You can see when you will switch on, you will see the 4 points on its screen while collecting, recording the passport information like village, tehsil, block, you are anywhere you are, which is must for every explorer. In exploration sense or survey or exploration, if 4 satellites are above, 4 satellite points are showing in your GPS screen, so then you can, if GPS showing only 2 points, don't take the GPS reading. When the 4 points are there, you wait for 2-3 minutes or 5 minutes for standardization, and definitely you will see the 4 points. Means 4 satellite should be above that point where you are taking the GPS coordinate, because in satellite or in GPS orbit, 24 satellites are there. They are moving east to west. They are moving there. But you will take some time. You are switching on the GPS, and you take some time for standardization. And when you will see 4 points are there, mean 4 points mean 4 satellites above your standing point, then you can take. One should verify recorded data with reported data, latitude, longitude, altitude. If you have the doubt, you can, maybe some satellite, maybe some problem is there. So accuracy is meant to know human GPS instrument error. So you can verify. GPS allow farmers to accurately navigate specific location in the field, days, month after days, month, all these things, to collect soil sample, monitor crop condition. Now people using GPS in different tractors also to monitor their crop. So you can here see this is the picture I have taken from this Google. Basically I have taken, and you see how Indian 6 satellite it is showing how they are moving. See, all in one direction. See, they are moving like that. Like so, this is Indian regional navigation satellite system, and worldwide you can see also you will be seeing how the GPS satellite is orbiting. So this is very important. Now the methodology used in PGR data analysis, where we are focusing here how to analyze data, how to analyze our PGR data. So we should know about the methodology part. The methodology for analyzing gap analysis, for georeferencing, points to grid analysis, variability mapping. So what we are using, see, analyses are based on the site collection coordinates, latitude and longitude, and additional attributes of point data. Means what, how many other columns are there in point data. So the points represent location where a specimen, example herbarium, passport data, gene bank data was collected. Herbarium we have collected from any area, that we call spatial. Spatial data means herbarium collected from some part, some place, that called the spatial data, and we can take those spatial data like the latitude, longitude, altitude from that ground. So we can link this herbarium samples collected from that particular area by using geocoordinates, and where any observation of the presence of a specific biological unit, for example, species, land range, genotype was made. So we can correlate those things. Georeferencing we can use those points. We can use in georeferencing. I will show the practical also. I will show this map also, how we are using this georeferencing. Point to grid analysis. Suppose we have the collected samples in point, so many points. People are not understanding on those points. So we can make, if we want to make the diversity richness map, we can use the point to grid analysis. Simple, we can use circular, we can use neighborhood method. Species richness, we can use how many species of brassica is there, how many species of sesame, wild sesames are there, and different variety also. If different names are here, we can use diversity indices, Shannon diversity. So Shannon diversity also we can use here for mapping. What is the value of Shannon diversity in that particular, based on species and collection. Variability mapping using static methods. So variability, we have the static method inbuilt in our Deva software. Eco crop modeling predicts the adaptation of a crop for a selected area. We can make the eco crop modeling for habitat prediction, actually predict the adaptation of a crop for a selected area. So this is very important. And in generalized, we develop, or all colleagues, we develop this methodology for how we can prepare crop diversity map. So we have the passport data, crop catalog data, national gene bank data, conservation data, we have crop information data, secondary sources from, we are taking from IRA, also from other sources, published literature, we are taking this data. GPS data available in data. GPS data, some data already available in database. Screening of data and input of geo coordinates. So because I will show that in next slide, how we have the without latitude, some data without latitude, longitude, or some data with degree, minute, second. So we are taking from Google or Gadgeter for published literature, or in Survey of India maps, district and state layers. We are using then if we have the Survey of India maps layer, shape layer, not physical layer, shape layer, district, state, and GS system is there, then we can prepare geo-referenced map. This is simple application. If we have the data, for example, we have the 10 accession, 50 or 100 accession for any crop, and we have the district, say state shape layer and GIS system. We have the software here. We can map geographical map. This much of it. But if we want to integrate in GIS format and data analysis, so we can identify some more things. GIS georeference map plus topography, environmental variables, identification of priority area for collection, then field data, current scenario, input from crop space expert, then we can develop this crop diversity map. So if we have all these resources, database, all other resources, layers, environmental data, we can develop this crop diversity map. So for example, we can discuss about the diversity, because diversity is so our student also ask about the, sir, we want to know about the how we can analyze diversity, particularly sand diversity using DivaGIS. We want to. So diversity is a mathematical measure of diversity in a community, plant community, any community. Diversity indices provide more information about the community communication. That simply is the richness, the number of species present. They also take the relative abundance of different species into account. If one species, if any community only 5 species is found, and one species dominant is the 95, another 4 species, one is 2, another is 1, total is 100. So dominance, relative abundance also it counts. Very, very pertinent index is that not only counting the total number of species, but the abundance also, dominance of that particular. I have given the example of the, for example, 100 individuals are there. Now total 5 species are there, and 100 individuals are total are there, and one species has the dominance 95 individuals, another 2, and another 3, 1. So you can relate this example, and so it counts the relative abundance. So diversity index provide important information about the rarity, commonness of species in a community, plant community, or you can say grassland, plant, agriculture. The ability to quantify diversity in this way is an important tool for biologists trying to understand community structure. That is the part of how the sand and diversity is increasing or decreasing. Spread distribution, assessment of species richness. DivaGIS is useful for mapping point data into grid analysis using this measurements example. Diversity can be calculated using sand and mineral diversity index. This formula you see in the website or everywhere you will find this formula. H is equal to minus summation. This formula you will get. But for simplification, this we have made this one. This C. H is equal to minus summation Ni upon N into natural log Ni upon N. And what is the Ni and N? This is the very, very important. Only you can say 3 values are here. Only 3 values. H sand and diversity index, Ni individual of a species. Whatever individual, I mean any species. For example, Oryza satava is there. How many individuals are there? 5, 10, 20, 100, 200, for example. And total individual all species. So 5 species are there. I already given the example. Suppose Oryza satava is the 95, for example. Another is, for example, another is 2, another 3 is 1. So you can see total individuals all species is 100. So you can calculate 95 divided by 100 natural log, value of the natural log into Ni upon N, same. So this is very, very simple, but the software is what is the interesting. The DivaGIS, any software, this formula is inbuilt, and through this the inbuilt formula you can make the sand and diversity map. Diversity map you can make. You can show the diversity values. So this is important. Now how the grid is working, because suppose we want to go fine grid survey. So we will take only small grid, small area. Grid in the sense of the grid means small area. You can say grid for this digital purpose to map the any diversity, any diversity or any features. So we have the grid technology to show in better on large way. And broad survey, we can go to big area. So in this example, you see grid has an extent which refers to the geographic area it covers. Mean grid is covering some geographic area. Grid is rectangular. It can be defined by two opposite corners, the upper left and this is simple. In case of the latitude longitude system, a resolution of one that is cell is 1 into n. Means that is 1 means 1 degree. So for example, 1 degree is equal to 111.35 kilometer. So 1 degree into 1 degree means 111.35 into 111. That is the 1 degree. And after 111 kilometer, the degree will be changed. So that is interesting. This is part of the basic question, how the degree is changing. Because India is covering from 7-degree latitude to nearly 37-degree latitude from you can say Kanyakumari to our top JNK, 7 degree to 37, and longitude from Gujarat to Arunachal, from you can say from here 66 to 98, in between that is the longitude. So you should always remember this. This is the latitude, longitude of the area of the country. So the value assigned to each grid summarize the characteristic or condition of a special variable map layer at that location. What is the in that location? We can understand based on the grid map. GIS data on the other hand is a relatively new way to describe geographic space and its relationship. So for fine grid survey, we can go one grid only. For broad survey, hundreds of grid we can use. Here you can see, you people might have been seeing this is part of passport data sheet. This is for presentation. This is not complete. See, this is not complete. Here I have given one example. Suppose you have this type of data, foxtail minute, for example, from any area, Andaman, Anandpur, latitude or longitude is there, but data is not there. How? This is the part of practical. This is the part of your practical. You have data. You can take, suppose you can take your nearby your house. If you are joining from your house or your village or your residence, you can note down 4 or 5 points or 10 points from nearby your house or nearby your area. Anybody is joining from IRA, they can take 5 to 10 points from different GCP, we call them, ground control points. Suppose we should use always GCP, ground control point. I mean, we should remember those, or we can identify that permanent feature. For example, the left hand corner of the IRA library and right hand corner of the IRA main gate, like that, and left hand corner of the back gate of the IRA, like that, and agriculture physics, the corner of the right corner of the agriculture physics building, NRL building, like that. You can make your point. Like this is the part of the practical, and there is nothing latitude longitude. You can take from the Google, find latitude longitude, many sources. So many sites for taking the Google Earth Pro. Also, you can use so many things you can use, and you can take always for analysis. For teaching purpose, you can use degree, minute, second, no problem. For teaching purpose, we can use. But for analysis purpose, always take the decimal degree. Means like 14.467 or 78.824. You can take decimal degree. It will work in your GS environment. That is the main. This is not okay. This degree, minute, second is the wrong. No. Degree, minute, second is not the wrong. But your software, sorry, our software or your software is taking only decimal degree, because they meant such software, they will take 2 points. Because earlier, binary, how the computer developed, binary system, 0, 1, 0, 1. You count anything, the 0, 1, 0, 1. Like that, binary system, we started during computer classes. So you can have the how, anybody, because you have not the permission to ask the question. But how you will get the latitude and longitude from any area, you can go the Google site and you can write there, find latitude, longitude, the map will come. You write their name. For example, you want to search Delhi, you write Delhi, India, and you click on that right side button, and it will show the Delhi, India, Delhi, capital of India, Delhi, it will show. And then you can zoom left, right corner, where is your point? Then you can zoom there and you click button is there. You can click and you will get the latitude, longitude, degree, minute, second, and decimal degree. You take the decimal degree. This is the first point of the practical. This is because your data is not there, how you will prepare that. So see, here you can see, we so many time we have discussed about the Diva GS, because this is the, you see here, analysis of diversity using Diva GS. Diva means diversity analysis, not any any different abbreviation. Diva means diversity analysis, that is GS. They developed. This is developed by the FAO funded one group of scientists. They developed this software for PGR scientists, PGR community, and mainly the professor Hisman. Now at present he is in California University. They developed this software. So we are working. You people can download. I can hear this. So you remember this Diva.js? You know the spelling, all these things. Now see, you know the how make your data. See, now you download this Diva.js from Google. This is not, because now everyone has the smartphone, Android. So you just write Diva.js in Google and you write Diva.js download. You will get the Diva.js manual, particularly 7.5 version. You download and you can read those. So you will see here, one icon is here, Diva.js red icon here. You click this one, it will open. This is your data. This window is your data view. You can note down. If anybody want to note down, see this window we call data view. This is not design view. This is the data view. Oh, you can. And design view, you can click design. Then you can add your this legend, all these things you can add later on. But this is your data view. In data view, you click on this Diva, D-I-V-A, Diva red color button. Automatically it will come when you will download. And another point is very important. You will get, because I'm not going to provide you this safe layer of India or safe layer of district, safe layer of, for example, MP, UP, Rajasthan. I'm not giving you. You can download from the Google site, Diva.js safe layer, and you will get, for practice at least, that is very important. And climate data you will get, country, world map you will get in safe layer, that is for practice. So you will see, when you will open this and you just make the right click, and here right click you will make and you add the layer. So you have, you see, you have the here project, see, sorry, some problem is there. See, this is the UC data layer, all these things. You can work on the data. First layer, you upload the layer, India layer. So this layer will come. This we call the India safe layer. This is the India safe layer. Here you can see we have the we have the district layer. Here two legends are here. One is India states, another is India districts. So green, all India, all this India districts are there. Okay, these are the India districts are there. In Indian state is there. Indian state in India district C. So these are the districts boundary layer. If you are working on only districts, you can separate each state based on the software. No problem. You can take this data layer and you just choose the MP. The Madhya Pradesh, you will get districts. And if you want to overlay the point data on this, well, the germ class collection data, any other data you want to overlay on this map, so you should have the Excel sheet. You should have the Excel sheet in with the latitude, proper latitude, longitude. And if in your Excel sheet in your desktop, you save that Excel sheet in desktop, and you import from data layers. From data layer, you will import and you will import this layer, and you will have the now two layers. One is India state layer, another is India district layer. See, another see. Now you want to see, you want to map this now you have the two layers. You see, one is the Indian states, another is a point data of any particular crop. See, so many points are here. So you can have the geo reference. You can call this geo reference map. You can call this geo reference map. And see, and after this data view, we can make so many maps for different crops. You can change this color. You can change this color. So yeah, now see. Now you can see. Here you can see different colors. You can see here. So different. We have the one crop data, another one crop data in red color, another black color. Different agroecological zones you can overlay. And here is the 1, 2, 3, 4 layers. So it is depends upon you how you are working. So if you have any doubt, we can discuss in later on. We can discuss about how we can work on physically in this one. If you have the passion, you can work, and you can contact us if anybody has the passion or working on this GIS or remote sensing or natural resource management, diversity mapping. They can ask the question. I will discuss here based on those correct data in Excel form, Excel sheet. So we can prepare first map. You can see collection sites of brassica species. These all collection sites or GPS points, all these based on latitude, longitude from that area, that site. We have already some data we have already in our passport data, and some data we have taken from Google, Gadgeter, or published literature. We have taken so many sources that we call this geo-referenced map of brassicas, and we just taken this 5,350 accession because we have the the locality information, complete locality information of this much of number. So see, in PGR study, georeferencing can be defined as the act of assigning location to germplant accession of information. Very good. It is essential in GIS because all information must be linked to the earth feature. Because in your database, nobody is interested to look on those books, those database, lakhs of database, those nodes, and those gadgeted. They have not time to see what is the status of collection, what is the status of diversity, what is the status of collection and conservation. I will show that map also. See, but through the technology, GIS technology, remote sensor technology, you can see quickly, you can analyze, and policy maker, they can see quickly. See, and in this first map, I want to discuss here. See, without much knowledge, anybody can say, yes, this connected diversity is showing, very good showing where the brassica species are growing more. That is reflecting there. So desert part, not that much of collection. Is here also not that much of collection in Tamil Nadu, this area, because this is not crop of that area, so it's less number of collection. So based on this, here you can see only the point data. You can say the point data of all species. This map is showing point data or collection site of all species. But you can see, this is very interesting. In second map, you can see the speciousness in rapeseed, mustard, and flower. If you are interested for your PhD, for your research, 4 species at a time, you will visit, you will make your trip or exploration survey or visit not in other area where red is there. Because wherever red is there, even Manipur, I visited 2 times in Manipur. We have seen 3-4 species are there growing in Manipur Valley, 100 kilometer long Manipur Valley is there. So all the part, this is the Bharatpur area, Rajasthan, Western part. Here is the UP, Uttarakhand, 3-4 species there. If you are going for the 4 species collecting at a time, you will go this part. But if you want to collect only 1 species, any part you can. So if you want to, you have used data, 5,000, 10,000 data. If you want to work on, your desire is to work on trait specific germ plant. Because out of 10,000, we have the 100 accession with some specific trait. Or if we know the trait specific germ plant sites means area, location, state, district. So we can go there. Suppose we know the eastern, southeastern part of the UP, this part of, you can say, Bihar and this Jharkhand, this area where this orange and red color, both orange and red color, most of the trait is generally found this area. We can visit only 3 states. Why you can go to other states, so many states? For example, Brassica actually found in 29 states. Why we should go 29? So we should go only, this is helping in that planning also. So see, here is the trait specific, but traits identified by the our evaluation division and our crop expert, we can call it. If anybody is working on Brassica, they have developed the promising germ plant or trait specific germ plant, potential germ plant. They developed. We have just identified the sites based on technology. So this is the help of the students. See here you can see we have discussed why we have the using GS for georeferencing and identifying gaps in collection and consumption. Here you can see when where the red dots are there, see collected germ plant. Where the red dots are there, that is collected germ plant, but where the conserved germ plants, those are the collected and conserved. But some germ plants could not reach due to evaluation procedure, other procedure, disease, so many things. Here we can see how we can overlay any pulse crop here. You can see here one pulse crop, another pulse crop. We can see one is red color, another is blue color, and different agroecological zones. Suppose your target is to collect or to work on hot arid region. You will go here and here, this part, Rajasthan or Kutch area of Gujarat, this part you will visit, or you will make your trip in these areas. And here also, some part of this, very interesting, some part of this Maharashtra and this border of Maharashtra and Karnataka, this also come under hot arid. So you will go there and you will collect these samples from this area. So that's why we should know to which part we are planning. So in northeast, others different area, hot subhumid, warm parahumid, or some. It is up to your objective. So see, so that's why we are in agroecological zones. So here we want to know the. We have already analyzed the gap, already analyzed in P, and this we want to know the performance of the different P in relation to topography map, environmental variables. So we can make not one map, we can make hundreds of maps. If you know the technology and interpretation, we can make this map for soil types map. See, so soil types map, you want to go this desert area, you can take this range, this area, different you can Gangetic plains. This is the part of Gangetic plains, all these things. The rainfall also you want to know the highest in this Cherrapunji, looking different. Here this Cherrapunji, and now the Moisin Ram, the different name is the Moisin Ram is the highest rainfall area near to Cherrapunji, that we know. This is this. So this software also can based on, and here you will see some highest rainfall in Western Ghat also. This is a small map, you can see. Here the annual temperature also you can see. See this large area over India. large area with the high 20 to 25 degree temperature in average actually. So here you can see unexplored area identified. Eastern Ghats, coastal area of TN and northeastern region focus for. This is just for this. Another we have developed, we have worked on this Brassica nigra. This is very interesting crop. And in this you can see only in that time we have only 12 accession in our gene bank, black mustard, Brassica nigra, and we have collected only 12 accession part of the Rajasthan and Haryana, and we mapped those accession very interestingly. And our senior colleague, they characterize, evaluate this data, and you can see how they can map this. Another this one I can show you. See, and here we use the not the simple technique but neighborhood method we use. We want to show those grid in larger form. So here you can see diversity for richness value of oil content in Brassica. So we can go for oil content because anybody want to high oil content seed or samples or germplasm. So we can go this area, these two districts, and for this purpose, the number of mapping of diversity for number of siliqui on main fruiting branch. So we can visit this area. So this year you can see the diversity value. You can see up to 0 to 2 sennen diversity value based on the collection data or this oil percentage, all these things. And here this is the result actually. IC5630 donor for high oil content like that. Na? So see, this is the result. You can say how we are using GS technology, how you are using this GS technology in PGR diversity mapping and the variability mapping or characterized data, how we are mapping. So another example, see, wild rice. We have published this paper in using eco-crop model. See, here total of this accession we have taken for analysis and see the maximum accession from Odisha. So here we have given, people can understand this different species, Nuara, Officinaceae, Rufico, we have given different based on, this is not based on my expertise, but this is based on passport data available in our, you can say, collection database or gene bank database. Based on this, we have identified this, we have mapped these things, and we found maximum wild species. We can found this area, this yellow grade and orange grade. 1, 2, 3, 4 area we can focus to collect maximum number of species, 4 area. So we know this, we can overlay this district layer, we know these districts, we know this, all these things, we can propose our visit. This is the help of the GS technology. Suppose anybody want to work on wild rice species for PhD work, for research work, for any genomic analysis, they can visit these 4 areas. And for particular, this is the example of Odisha we have given. So see, this is for another Brassica tournifortia here, because we are giving the interesting result. Because see, wild Asian mustard, Brassica tournifortia, we have only 18 accession that we call Asian mustard also, because this is very interesting crop. And see, on the basis of area identified collection of trait spectrum about tournifortia, area number this is the value of the sennen diversity. So you can cluster of oil content. So we have used this technology. So this we can use this technology, no problem. We can use this technology in various here. Here I want to give the example of wild sesame. So when we analyze this, species, particularly wild sesame, we have found more maximum or most of the wild species found in peninsular region of the country. And you can also see when we have discussed with the crop expert, they appreciated this. Yes, most of the species, and here you can see only Sesamum malabaricum found in northern India. See, where everywhere this one color. And see, malabaricum found in peninsular region also. So you can see 1, 2, 3, 4, 5 species found. Now they have developed this wild Sesamum catalog also. So see, so based on the data, wild species, if data are correct, so we can make the very good map for the diversity analysis. See, so geographic speed is a map of collected wild species from different regions of India. So in a glimpse, we can say, yes, in northern India only found one species and another subspecies. We can say, now some people identify this subspecies also 1 or 2. But peninsular 5, 6, 7 species we are finding. So based on the GS technology, mapping science, point data analysis, quickly we can say, yes, where is the area where we can get the maximum species. Mapping of promising and traits was Solanum melongena also. So we have prepared like that. Brassica, we have prepared this. This is also great technology we used. Here we can suppose orange color we found only 2 places. So many people, expert, they appreciate this Solanum. Most of these we from this West Bengal and this Assam, this part. This is also part of the most of the part of the West Bengal, this Coochbehar area, all these things. So it is helping actually. This technology is helping to us to understand the data distribution, data habitat. What is the habitat prediction mapping? So we should know that what is the habitat prediction using crop models. We have the eco crop model inbuilt in DOGS, bioclim model, domain model. So we are using basically bioclim and this one eco crop model. So mapping and predicting the special distribution of crops and their crop quality is main challenge to habitat management. Habitat mapping quantify the relationship between species and habitat and is evaluated according to the species performance. It is basically, we are basically till today we are dependent on the for the climate data or environmental variables. We are taking actually data from worldclim.com. So climate data you people also can download from worldclim.com and DOGS site also you can download the climate data. So here we discuss about the sit area. So we want to know, we have analyzed the gap, analyzed the foxtail millet. We want to know what is the habitat, precious habitat based on the data. So we cannot validate this, but we'll validate this. So see, model predicts that most of the area, more than 40% of eastern area is the suitable habitat for foxtail millet. Foxtail millet needs prioritization for area for future germplasm collection. So we should know this Chhattisgarh, this Andhra Pradesh and Odisha, that belt also, Uttarakhand you can see, Northeast also, because high and very high, we can club those also. High suitable area, very high suitable, we can club. So see, most of this area, this crop is found, but not in this other area. And that the software is, sorry, that model is predicting more than 40% area is suitable in these eastern region area. So foxtail millet habitat suitability map based on current we have not changed the climate. We have the provision in bioclim model. You can change the climate 0.5 degree, 1 degree, 2 degree. We have not prepared 4 or 5 maps, but we just as a current climate.
Hello.
Which is due to 2000, and they make the first current climate, and all the people they are using this.
Hello.
This is
Sir.
based on this, not future. Ravi, any?
Okay, sir. Some technical issue. Okay, sir. Continue, sir. Yes.
So see future prospects, we can see in PGRST, GS technology could be of great use in the management of plant genetic resources, particularly in the following thematic area of studies. Integrated geospatial technology can be used in gap analysis, planning and execution of future exploration program at national level. And when our institute, we can call, when our institute is preparing national exploration plan, we are using all gap analysis data. Crop wise, we are going through collection and conservation, and from the KVK also, KVK help also we are taking. We are doing georeferencing also we are doing, and based on that we are developing national exploration program. Nearly 40 average 40 exploration we propose for every year for all over the country. So we are using geospatial technology and GenPlus password data information, remote sensing data, spectral signature and climate analysis can be used in diversity distribution mapping and prediction of diversity area for different crops and their species. Now we have to identify the specific area of diversity richness. So now we are using complementary technique. One technique is there out of you can say how you will use the only one or two sites. Based on complementary technique, we are using this complementary technique to identify hot spots, micro hot spots or richness area, diversity richness area of the particular region. Field remote sensing data could help in delineating crop relatives, because we have the crop relatives very precious germplasm. We should need a spectroradiometer to record spectral signature of target crop at point sites in the field or farm. Because now the spectroradiometer, they reduce the cost, and that instrument also is advanced. So we can use this instrument for field remote sensing, particularly for identifying different crop wild relatives signature. And we can use this for extrapolation of the area, because suppose where Cajanus wild species we got near Odisha, one part of the northwest part of the Odisha, we got the Cajanus, Cajanifolius and other species, wild species area. Based on the signature, we want to extrapolate that area surrounding the Chhattisgarh and Andhra Pradesh, where we could get the Cajanifolius species. So we are focusing for the field remote sensing and ecological parameters also. Quantitative parameters we want to make in this study. And hyperspectral remote sensing can be used in distinguishing and identifying crop variety of cultivated plant in future. This is for future, because hyperspectral remote sensing is so costly. So it is very tedious job to take the hyperspectral remote sensing data. And drone remote sensing technology could be used in disease mapping. That I already discussed. Disease mapping is very important. Pesticide spraying, monitoring of field crops to note the moisture level, growth condition, maturity of crops at temporal scale. Temporal means time interval. 15 days interval you can take, 10 days interval you can take. So this is very interesting. And in drone, we can use the different cameras also, different sensor we can use. But I want to flag this point here. ICR funded nearly 35 drones to all different ICR Institute, Krishi Vigyan Kendra. Drone, they funded 35 to 40 drones for the demonstration of pesticide and other nano urea to farmers. And so farmers can use in future this technology as a part of their cultivation practice. So that's why this time government of India also supporting drone technology to farmers with, like that lab to land technology, because lab very good technology, very high technology in our lab, billions of rupees people are using, but is that technology is going lab? So this is another one example. I'm not saying only one example. This is another one example. Drone technology, we can, many institutes, they are now showing to the farmers in their fields how we can use for spraying pesticide, nano area, or this water, some dry area. We can use the dry, because the capacity of tank is that tank is the 10 to 20 liter at a time we can use, and this is the remote piloted. We can take the training. Our office and any participant, they can take training for 5 to 6 days. They can use the drone technology. This is one of the advanced technology which is going to hand off the farmers. And what we use, the farmers of big, you can say the land holding, large land holding. They have 500 acre or 600,000 acre. They are now thinking to buy the drone for their agriculture monitoring, crop monitoring. High spatial resolution, 0.41 meter emotion data can be used mapping of disease system in crops and their wildlife at fine grid level. One big field is there. For example, in IRA, we have the big field is there. But due to high resolution satellite data, we can use only 50 meter or 10 meter, 20 meter area for disease symptoms identification. Because one thing I want to explain here, our eyes only can detect, this is the thumb rule, our eyes can detect only 20 to 25 grades of color, 20 to 25, or you can say 30 grades. But satellite can identify more than thousands of grades, 500 to thousands of grades, colors. They can gray levels, we call that gray levels, radiometric resolution. They can identify thousands of distinguished colors. So that's why we are taking, because if any leaf, once disease is there inside, that disease is appearing, but leaf is appearing outside green. We are saying no, this is the green, this is a healthy crop, but inside the disease is there. Satellite sensor can identify this is the disease plant. So this is the last suggestions. Thank you, Ravi. Any questions?
Thank you, sir. Thank you, sir. It is very highly informative slides, and your presentation is such a long-lasting and very highly informative. And dear participants, I am requesting you, if you have any doubts, you can interact with the resource person, speaker.
Hello, sir. Hello.
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 Yes.
Myself, Dr. K. Gupta, Jabalpur. Sir.
Yes.
I ask one question. Can I identify particular disease for this technology? Means any disease, particular disease, rust of heat.
No.
Blight of rice.
No, we have the crop expert together. We can take the help of the remote sensing data and spectral field remote sensing. We know the relationship of disease and crop expert. We can identify easily. That will give the symptoms. That will give the digital value, and that study also already published by the NRSA. They published this study, so we can say this disease occur in this crop, for example, wheat and rice. This is well known. So we can just what is going inside the structure of the cells, chlorophyll, mesophyll, texture, all these remote sensing identify. what is happening.
Thank you very much.
Visualize.
I understand.
Thank you. Thank you.
Thank you very much, sir. Thank you very much, sir.
Okay, welcome.
And anyone, and any participant, if you have doubts. And sir,
Excuse me, sir.
Yes, yes.
Yes, sir. Very good evening, all of you. Thank you so much for your wonderful presentation. I have a question. You know that NBPGR have the more than 100,000 wild accession for the rice germplasm. So your team have screened for the disease resistance or any abiotic resistance or insect resistance. Can we get that information accordingly? We can order to isolate or map the gene. This information available in NBPGR?
Sir, it is, sir, in next classes, evaluation scientists.
They will tell about this.
And some are already have, sir. They are already registered under INGR varieties. Trait specific germplasm also they registered. We will get, sir, in database also there in PGR database.
We can check on database.
Thank you.
Thank
you, sir.
Sir, and someone posted in chat box, what is the cost of commercial GIS commercial software?
The ArcGIS, suppose anybody want to full operation of commercial GIS software, that it will range from 20 lakhs to 40 lakhs. 20 lakhs to 40 lakhs. ArcGIS now they have reduced the cost. Earlier it was more than 40 lakhs. Now they have reduced the cost of 20 to 40 lakhs. They have divided in different units. They divide the software, and for the complete package, it is more than 40 lakhs.
And sir, one question came from Dr. Reena Mehra from ICARDA. In the NDY and RBY, which were, how it is different and which one is more effective and less in cost? Even sir, you can see in chart box also, sir.
No, NDY, yes, that we can. NDY is the better than RBI because we are taking here we are taking NDY actually near infrared and infrared band, and in this case we are taking only RBI, only 2 bands. So because near infrared has the high reflectance, 45 to 50%. So most of the people using NDY. NDY is better to identify any you can say vegetation, discrimination of any vegetation or agriculture, crop health. So NDY is better than RBI. So formula I have presented. That formula is there. But why we are using NDY? Because see the NIR has the highest reflectance value, 45 to 50%. So that's why.
And anyone, if you have any doubts, you can interact with.
Still, we have 8 minutes, no problem.
Still time is there. Yes.
Time is there.
I hope there are no other doubts. And sir, thank you once again. Thank you very much for your valuable and informative presentation on GIS tools in PGR management. And thank you participants for being a member. And please kindly, I am requesting you, please switch on your videos. We will take one group photo. Please, you are requested to switch on videos. And tomorrow we have a very good presentation is there. That is Introduction and Exchange of Plant Genetic Resources by Dr. Vandana Tyagi. And another one is Application for Import Permit, STMA, and some other principles and procedures of germplasm exchange. Actually, these are very important and highly useful in our research work. How to import the material and how to getting the applications and what are the procedures, you will get it. It is, and please attend the tomorrow classes. That is highly informative. And please, Ankur, please kindly take one photograph.
It's done, sir. Done, sir, from my side. Thank you.
Thank you very much. And thank you for attending all the participants. And please kindly attend the, and daily we are note down the attendance of the particular candidates. It is mandatory. Thank you very much. Thank you. Have a nice day. Tomorrow sharply we will meet at 9:30 a.m. Thank you.
Thank you, Ravi.
And thank you, Vijay Mandalji, for facilitating the facilities of Zoom.
Thank you, sir. Close kar do meeting?
Yes, sir. Yes.
Okay, sir.
Thank you. Thank you.
Thank you. Thank you.